which uses Nafion tubing and liquid water. But the instruments that you have there probably have the previous humidifier that has a porous ceramic media in it. And then there's a column of desiccant, in this case silica gel. And then I've put a CO2 cartridge in the instrument. So we use these little cartridges of compressed carbon dioxide, and that's what provides the carbon dioxide to the leaf. When the instrument starts up, you see a screen like what's displayed here. So this is asking us which aperture is installed in the instrument. This is in reference to the aperture here where the gaskets are. On my instrument, this is the 6 square centimeter aperture, the largest one for the fluorometer. And then it's going to ask us what we want to do with the fluorometer, whether we want to use it as a light source or we want to do fluorescence measurements. And the experiment that I think I'll do today, as we're working through things, is we'll do a light response curve where we'll change the light intensity. And at the same time that we do the carbon assimilation measurement, we'll do the fluorescence measurement so that we can look at maybe the relationship of ETR or phi-PS2 to light as well. When you're starting the instrument at the beginning of the day, the first place that you would go once you have the chemicals in the chemical column, when the instrument turned on, is to these warm-up tests. And I talked a little bit about these yesterday, where the instrument will go through and check its operation and make sure that it's working correctly. I'm not going to do them today. I know that this instrument is working correctly. And so the next thing that we would do would be go to the environment, and we would start setting up the environment that we want to use to make measurements. And so the environment that I'm going to use, we're going to start by setting the flow rate through the chamber. If you remember yesterday, flow rate has to do with the signal that we see and how quickly the chamber stabilizes. So in this case, I'm going to use a flow rate of 400 micromoles per second. I'll show you how much leaf area I have in the chamber and explain why I did that in a moment, but I really only have my chamber about half full. And so the default flow rate for the fluorometer is 600 micromoles per second because I'm only really using half of the area. I'm going to decrease the flow rate a bit. And that's going to give me a bigger signal. And then I'm going to use a slight overpressure in my chamber. And by slightly overpressurizing the chamber, if I were to happen to have a leak, it would force air out that leak so that whatever I'm doing around the chamber doesn't affect the measurement. Now, my instrument's fitted with polymer gaskets, and they tend not to leak in the first place, but it's still good practice to set this to maybe 0.1, 0.2 kPa. Next, we'd set the humidity that we want to work at. I gave the rule yesterday that we want to be between 1 and 1.5 kPa for BPD. That's generally where we try to start. And so I've set my instrument up to control the vapor pressure deficit of the leaf at 1.5 kPa. Next, we're going to pick a CO2 concentration to work at. And I'm going to go into great detail in a few minutes about how we choose these CO2 concentrations, whether we control on reference or we control on sample, what that really means, how we might pick this number. But to start my measurement here, I'm going to control on sample, and I'm going to pick something that's close to ambient, maybe a little bit above it, because this plant's been setting inside all day. I turn the fan speed on. For the most part, you're going to operate at whatever the default fan speed is. That's 10,000 RPM for most of our chambers. We talked yesterday a little bit about sort of some special cases here for broadleaf tropical understory things or broadleaf tropical trees, where you might see the stomata start to close in response to the turbulence in the chamber. And that's when you would turn this fan speed down a little bit. The green bars give us the recommended operating range. And generally we work at 10,000 unless we know we need to otherwise. And then I went ahead and turned my temperature control on at a nominal ambient value. And the part of the instrument that I'm controlling is this piece right here. So this is the exchanger. And when I showed the mixing fan yet, it's coupled to the air inside the chamber, which is coupled to the leaf. When we go to start our measurements, I'm going to switch from controlling the exchanger to controlling at the current leaf temperature. And when I talk about how we do this particular measurement, I'll explain why that is. And then I've set my light source. So you can see we have a number of light options. And so when you first go in here, it might start with ambient. This is related to the quantum sensor on the outside of the instrument. That's measuring the light outside of it. And you can see, as you might expect for some place that is at night, it's very dark in the room that I'm in. It's 2 ppfd, even with the lights on. So this panel, where it says fluorometer, is where I set the light intensity being provided by the fluorometer. And so I've set it to provide equal amounts of red and blue light. So this color spec here says R50. That means 50% red, and it'll automatically make the other 50% blue. The default that we normally use with the instruments is R90. For what we're doing today, it's not such a big deal, but I would start with the defaults. And then I've set my light intensity that I want to start at, and I picked 1,000 micromoles per meter squared per second. So 1,000 ppfd. That's half of nominal full sun. And I think that should be something that's maybe close to saturating, if not a little bit below saturating for this plant. And then I went ahead and set some fluorescence parameters. Let's move this thing. So I chose modulating rates. If you remember my fluorescence talk yesterday, this is how fast the light wiggles on and off. And then I know that this plant is a highlight adapted C4 plant. And so I'm going to use this multi-phase flash protocol to do my fluorescence measurements during the curve. And right now, this is just the default settings, and this is a fairly reasonable place to start for these kind of measurements. So the next thing I've done in setting up my instrument, because my leaf that I'm measuring, I don't know if you can see this well or not, doesn't completely fill my chamber aperture, right? This is only about 1.2 centimeters wide, and my chamber aperture is about 2.75 wide. Under constants in here, I made sure my leaf is going down the middle of my chamber aperture, and then I input the leaf width. And when I do that, the instrument will compute the actual leaf area in the chamber, okay? So that's very critical if your leaf isn't going to fill the whole chamber, to tell the instrument how much leaf area is in there, either before you make the measurement, in this case, or do it after you make the measurement in the data file. And then, if we go to measurements mode, we can just set and watch this thing. And it's not doing much. Let's hope that this turns around. But right now we see an assimilation rate of about 0.6, and it's very noisy because we're at sort of the noise floor. And a stomatal conductance that's what, 0.015 or somewhere around there. That's a very close stomata. And so hopefully, once this thing sets in the light for a while, that it'll start to wake up. I don't know that it will. They cooperated yesterday, but it might be too much to ask it to do the same thing for another day. We'll see. But while we set and wait for it to wake up, what we're going to do is I want to give you a talk about sort of the different ways that we apply the instrument to get different kinds of measurements from it, and the logic that we use to decide how to set it up to make a particular measurement. And so I'm going to stop sharing this, and I'm going to switch to this guy. Okay, so yesterday, oh, I'm one slide ahead of myself. Okay, so let's see. A very common situation that I end up in at Li-Cor, and all of our scientists end up in this situation, is often at the end of the field season, we'll get calls or emails, inquiries from people, and they'll ask us, Jason, I took my instrument to the field and I collected a whole bunch of data with it. What do I do with that data? And my guess is that some of you are here today because you haven't collected any data yet, and you're wondering, what do I do with that data? And you're in the best situation, because what I really want people to do before they take one of these instruments to the field is I want you to sit down and think about how the data you collect with the instrument's actually going to help you answer your research question. It's very easy to push buttons on an instrument like the 6800 or the 600 and get numbers back from it. But what those numbers mean, they have a lot to do with how the instrument was actually used to collect the data, right? I'm going to show you some parameters today where the meaning of the parameter is totally different depending on how we set up the instrument. That's a good thing. And as we go through this, what I'm going to do is I'm going to present sort of a series of measurements that if I were working with new plant material and I was designing an experiment, the first thing that I would do is go make this series of measurements on some subset of my treatments, on some subset of my plants. Because these measurements that I'll explain as we go through this, they'll tell me sort of how my plant responds to the environment. It'll tell me how I need to set up my instrument to actually make the measurement. It'll tell me information about the best time of day to go collect that data. I know that was a question we had yesterday. Okay, so we collect a little bit of data using a number of different experiments. That's going to guide us in how we set up the instrument and what the best data to actually collect for the research question is. So we talked yesterday and I presented sort of how the instruments work from a first principles approach. And I said, you know, we measure a number of things. It's not carbon assimilation or transpiration. We measure things like gas concentrations, temperatures, pressures, flow rates, right? And from those, we compute 4 basic parameters: the carbon assimilation rate, the transpiration rate. I have shaded gray here because it's really the apparent transpiration rate. The stomatal conductance. Typically, we think of stomatal conductance to water vapor, but this could be stomatal conductance to CO2. And it can be CI, right? So these are our basic physiological parameters that the instrument computes. Now we can take the instrument and we can apply it in different ways to get special versions of these. And from the way that we apply it, we can derive other parameters. And I'm going to talk about a bunch of these, but before I get there, I want to say that pretty much everything that I talk about for parameters that we can get back from the instrument is documented in this application note defining the value of photochemistry parameters measured by the Li6800. And so when I mention things as we go through and you think about them later and you say, okay, well, what did Jason say about this? Or I want to know more about it. If you go find this application note on our website, the link is down here. This gives you the most commonly measured parameters from the instrument. It gives you a description of what that parameter is. It gives you the units the parameter is measured in, and then a whole bunch of literature references that talk about the underlying theory or underlying physiology behind that parameter and best practices for measuring it. So this is a really useful document. So let's just dive right into this. So fundamentally, when you take an instrument to the field to do leaf level physiological measurements, there's really only 2 ways that you can possibly apply that instrument to make the measurement, right? And we call these two ways, we call them response measurements or survey measurements, right? And a response measurement is a measurement where I have environmental control of the environment the leaf is seeing. I'm going to set all of those environmental parameters, one of them, and I'm going to look at how the leaf responds to that changing parameter. Okay. That's a response measurement. That's the kind of measurement we can make with a 6800 because it controls carbon dioxide and water vapor and things like that. On the other end is what we call a survey measurement. Okay? And a survey measurement is where I'm going to take my instrument and I'm going to set the chamber environment up to perfectly match what the leaf was already seeing. I'm going to put the leaf in there and I'm going to take a picture of what that leaf is doing under the current environmental conditions. Okay. So response measurement is about measurement where it responds to the environment and look at what the leaf does in that. And a survey measurement is always sort of instantaneous, what it's doing at that moment in time. Okay. There's a role for both of these kind of measurements. One of them is not right, one of them is not wrong. I generally prefer response measurements because they're highly constrained. You can generally make fewer of them to be able to draw a scientifically valid conclusion about how the plant behaves. And I'll actually show you some data that supports that statement as we talk about the 600. They're key to one of the data sets that I would tell you that you should always go collect when you're starting an experiment. So survey measurements should be incredibly fast. The only thing that we're waiting on is the response of the instrument. If we've done a good job setting the environment in the chamber up, and the response time of the instrument is really quick when we clamp it on a leaf. So the data that I'm showing here, the blue line, is I just logged the instantaneous assimilation rate at fixed time intervals after clamping a leaf in the chamber. And I should say that this data is on corn, which was measured under full sun conditions in the late morning in a field in Nebraska, right? And so, and then I just fit a simple exponential decay to it. I mean, more or less, that's what the data follows. There's some less exponential decay. And you can see that at the 20-second mark, 22-second mark, we're at 95% of the full response. 30 seconds, it's completely stable. In this data set, this is one measurement from a much lower for us to perceive the world like a plant perceives the world, right? The key to getting this kind of quick data with a 6800 is understanding the environment as the plant sees it. And we're going to talk about that as we go. When we don't perceive the environment like the plants, and we put it in the chamber, now we've changed its environment and we have physiology changes with changes in the environment. But typically, if you're clamping a leaf in the chamber to make a survey measurement and it's taking 5 or 10 minutes for things to get stable, it's because you dramatically change the environment the leaf is seeing. It's not really a survey measurement anymore. The first thing that we have to think about is the light environment, right? So light matters more than is near instantaneous, right? The problem is that leaves see light very, very different than people do. So a plant has a very flat spectral response to light. There's some variability to it, but the reason that we define photosynthetically active radiation the way that we do is because if you give a plant a photon that has a wavelength between 400 nanometers and 700 nanometers, each one of those photons has the same capacity to do photochemical work, right? I showed that idealized energy diagram yesterday, and we saw that both the high energy blue light and the low energy red light had enough energy to get chlorophyll excited to the first singlet state, right? Now we all know chlorophyll doesn't absorb outside of particular regions in the red and the blue, but there's accessory pigments that are associated to the antenna complexes that expand the amount of usable light that a plant can use. If you standardize to absorptance, it turns out you get the same quantum yield for plants grown under broad light spectrums if you measure in green light versus red light versus blue light, right? So plants are fundamentally photon counters. They don't care what that photon looks like. People, on the other hand, we have wide dynamic gain control on our vision. So I'm sitting in a room. We've already established that room is at 2 ppfd. That is 1,000 times lower than full sun intensity. And I can see just fine in here. It seems a little dim, but it's not that dim. My plant's completely asleep because it's too dark for it to do anything. If I go outside in the middle of the day, yeah, it seems brighter, but I can see just fine. It's not that bright, right? And that's because my eye is adjusting to compensate for that. Leaves don't have that. So when we want to make a measurement, a survey measurement, we need to characterize the light environment that the leaf is seeing. And we need to understand that there's two components to light that really matter if you're a leaf, right? And those components are the fraction of direct radiation and the fraction of diffuse radiation. So direct radiation is just light that has a directionality to it, right? So if you look at something like this, this hops canopy, there's a maple tree in there too, but this canopy, and it's under a clear blue sky like this. Almost all of this light is direct. And you know that it's direct because if you look at the shadows in the canopy, there's strong contrast between the shadowed parts and the illuminated parts. And those lines that define the edges of the shadows are very crisp, right? If we were to look at the same canopy on a cloudy day, right? Clouds are diffusers. They scatter the light all over the place. Diffuse light is coming in from all angles. There's not one predominant direction to it. And on a cloudy day or in a dimly lit room like I'm in, the shadows have blurry edges. They're not well defined. There's not strong contrast between the light and the dark. And that tells me the light is predominantly diffuse. So what does that matter? Well, if I take these leaves that are setting here, all of these fully sunlit leaves, right? And they're all setting at slightly different angles, right? Well, if the sunlight has a directionality to it, and my leaf is setting like this, and the light's hitting it, right? A beam of light is now spread over a larger area relative to a leaf that's setting like this. And that means that while they're both uniformly lit, this leaf is seeing more light than this leaf, right? That's because of direction of the light and leaf angle. And those differences in light intensity can be huge. It could be an order of magnitude difference, right? On a cloudy day, it doesn't matter so much. So when we go to make a survey measurement, we're trying to capture the light intensity that our leaf is currently in. And so we have two ways to go about doing that, right? So one methodology is that we use a chamber that's clear, right? Then we have the ambient light shining through the chamber onto the leaf, and we can just clamp the leaf in our chamber and we can use that ambient light. And that works great unless your leaf is setting like this, and then you go clamp on with the instrument. You have to hold the instrument like that, because if you change the leaf angle, you change the light intensity. And with a clear chamber, we also have to think about whether we're going to shadow the leaf when we make the measurement or not, right? So the edges of the chamber are opaque, And we want to make sure to pick a leaf that when we clamp on it, we're not creating shadows from the edges of the chamber. So the other methodology, and what I prefer, is to use a light source, like the fluorometer in this case, and to use the quantum sensor on the instrument, right? This thing is a cosine corrected quantum sensor. And that just means that it sees light the way an idealized leaf does. So there's a little white diffuser in the middle, and that sensor is measuring light incident on the plane of that diffuser. And the sensor is filtered such that it counts every photon from 700 nanometers to 400 nanometers of the same, just like our idealized leaf. So if I want to measure this leaf, I can hold that little white diffuser in the same plane of the leaf. I get a measurement of the ambient light. I can set my light source to it on the instrument, and then I can clamp on the leaf, and it doesn't matter what I do. I want to show you how big these leaf angle effects can be. They can add significant noise to the data if you're not careful about them. So this is data that I collected with an Li600, the Porometer fluorometer, and I'm going to talk about it in some detail in a little bit because it's a new instrument. I'm very excited about it because I've wanted to collect this data set for many years, and it was too burdensome with the 6800 to really do this with any sort of sample size and with any sort of speed. But the 600 was like that. So I have two parameters plotted here. I have phi PS2, that comes from fluorescence. That's a measure of the operating efficiency of photosystem 2. And it is directly linked to how much carbon assimilation the leaf is doing at that moment in time, and directly linked to the light intensity, okay? And then I have stomatal conductance to water vapor, so how open the stomates are. And then I collected this data on the same plant canopy. This was a tomato canopy in my garden. And the open symbols are from a densely cloudy day. The light was perfectly diffuse. You could not find a shadow outside. That's how cloudy it was. And then the solid symbols are from a clear blue sky, a sunny day, strong shadows, very crisp lines, all direct radiation. And what I did was I took my Li600, and I have one here, and I clamped on my leaf, and I made a measurement, and then I turned the instrument 45 degrees to the south. I'm in the northern hemisphere, so the south is where the sun is. And then I turned it 45 degrees to the north from normal, right? And the data that I have plotted are the initial value, and then the range when I went south, or when I went north, and when I went south, right? So this is light intensity. And you can see on a parameter like phi PS2, this thing responds instantly to light. On my sunny day, I changed my leaf angle. I went to the north where the light intensity went down, and there's a massive drop in phi PS2. And then I went to the south towards the sun where the light intensity goes up, and there's a massive increase in phi PS2. No, I have that backwards. The drop when it went up, the increase when it went down in light, right? You can see these variations are huge. 45 degrees is a relatively small change in leaf angle. Now, when I do that same experiment on a cloudy day where the light's diffuse, it's being scattered all over the place. Note, there's no change in light intensity as I rotate the leaf. And thus, there's no change in phi PS2. Now, when we look at something like stomatal conductance, right, this is slow to change. Phi PS2 changes instantly. Stomata respond on a tens of seconds to tens of minutes time scale. Because these measurements were done quickly, even on the sunny day where there's a massive change in light intensity, there's virtually no change in the measured conductance, right? Because I'm doing this measurement quicker than the physiology can respond. I hope this makes sense. This is a really key thing to get around because it's very different than the way we perceive light as humans. We want to think like a plant. So that's light. In a survey measurement, the next thing we have to think about is the carbon dioxide concentration, right? We have a leaf that's hanging out in the atmosphere at whatever atmospheric carbon dioxide is. So that nominal value is about 410 ppm for the well-mixed atmosphere, depending on season and where you are on the planet. That 410 ppm may have nothing to do with where you are. In that, I've measured here in Nebraska over the diurnal day, over the course of the day, differences of over 400 ppm in the carbon dioxide concentration. Inside the corn canopy in the middle of the day, when it's very sunny and everything is well irrigated and photosynthesizing away, I've measured atmospheric CO2 concentrations below 300 ppm. In that same canopy, in the middle of the night, when things are respiring and there's very little turbulence to force air out of the canopy and into the atmosphere, I've measured concentrations over 700 ppm. So we don't really know atmospheric until we measure it. We're going to do survey measurements. One of the things that we do with the 6800 is we'll turn the CO2 mixer off, and we'll set the soda lime to bypass, and then we'll step away from the instrument so that we're not influencing it, and we'll see what the reference concentration reads in a couple minutes. That should give us a good measurement of the atmospheric concentration. Once we know what the atmospheric is, then we have to decide whether we're going to control at CO2 reference at that value or CO2 sample at that value, right? We saw on the instrument that we had both options. So let's say we decide that atmospheric is 400 ppm, and we set our reference to 400 ppm, and we go measure our first leaf, and it has a pretty good assimilation rate. It gives us a delta of 20 ppm. Well, okay, sample's now 380. I've measured my leaf at below atmospheric. Okay, maybe that's an issue, maybe it's not. Now we go measure the next leaf, and it's doing even more photochemistry, and it's got a delta of 40 ppm. Well, if I'm still controlling reference at 400, then for that next leaf, sample is going to be 360. Not only have I not measured it at atmospheric, but now I've measured the two leaves at different CO2 concentrations. There's a biological feedback between the CO2 concentration around the leaf and the observed assimilation rate, right? My preference for measurements is to always use sample CO2 control for survey. And I will set this at atmospheric, and then the instrument will vary the reference concentration either up or down to keep this number constant. And then every leaf that I've measured, I know I measured at atmospheric CO2, and I know I've measured them all at the same carbon dioxide concentration. Now, that's predicated on me being a careful scientist. And what I mean by a careful scientist is, in a survey context, when I go to put the leaf in the chamber, I don't want to breathe in the chamber, right? Because if I breathe into the chamber, my breath is somewhere between maybe 30 and 50,000 ppm for carbon dioxide. And there'll be this big spike, and the instrument will take forever to stabilize after that because it's got to flush it out, and then the CO2 control has to get it back. So the sample control works really well if you're careful not to breathe into the chamber for survey measurements, and if you're working outside in well-mixed conditions. Sometimes we'll do survey measurements in a shade house or in a greenhouse or someplace like that where there might not be a lot of turbulence. And then it's not uncommon to use reference control. And what we would do if we were going to use reference control is that we would measure a representative leaf or a couple of representative leaves, and we'd figure out about how big the delta would be, such that when we put a leaf in the chamber, we get sample at atmospheric. And so if we decide that atmospheric is 400 and our typical delta is about 25, then we'd set reference to 425, and that would get our leaves pretty close to atmospheric for the measurements. Okay, But using sample control is by far the most constrained method. We talked in detail about this yesterday because of boundary layer conditions. But we generally want to target a humidity somewhere between 1 and 1.5 kPa. It's likely that in Indonesia, because it's already humid there, that you're going to be at the lower end of this range. But somewhere in there is where we would start. If we find that changes in atmospheric temperature cause problems, then we would step back from the VPD control and we would try relative humidity control. And there you're probably going to want to be in the 60 to 75% relative humidity range. And then we have to pick a flow rate. And when I was showing you how I set up my instrument tonight to make my measurements, I said that I turned the flow rate down because I didn't have very much leaf area in my chamber, right? So that's the S here. If I cut my flow rate in half for the same assimilation rate, this delta doubles, right? I would like to measure the largest delta that I can. And so I showed this data yesterday where we talked about the relationship for a given assimilation rate, the green points, that if we change the flow rate, you can see how the delta CO2 actually behaves. And the relationship between this delta and the uncertainty in the actual assimilation measurement, right? So this uncertainty assumes 0.3 ppm peak-to-peak noise, sorry, 0.2 ppm peak-to-peak noise. And at 10, you know, I have error bars that are spread over a range of a little less than 1 assimilation unit. And if I turn my flow rate down to get a delta of 35, then my error bars are within the point that I've plotted here. Now, for a survey context, if I'm at a really low flow rate, then it'll take longer for things to stabilize when I put the leaf in the chamber. If I'm at a really high flow rate, then things will stabilize quicker. And so it's a trade-off between speed and signal. And so if this were the actual example that I were looking at to set up my instrument, for this data, I would pick a flow rate somewhere between maybe 300 and 500, right? This is a pretty good delta to measure. The error bars are pretty tiny. I'd be happy with that. I should say this data is from a 6400, right? And the story gets is better with a 6800 because this peak-to-peak noise spec is smaller. The final thing that we have to think about in the survey measurement context is what we want to do about temperature. And generally, it's a trade-off between how much power you can take to the field with you and how constrained you want the data, right? So temperature control uses the most amount of energy, right? So the more tightly I want to control the temperature, the quicker I use my batteries up. And in the 6800, we're pretty efficient about the temperature control. It doesn't use a lot of battery capacity, but it uses some. And so my preference for survey measurements is to set the exchanger temperature, the piece that I showed you that I was controlling to start with here, to whatever the ambient temperature is. And that generally means that the exchanger is going to cool just a little bit. And that's going to compensate for heat being generated by the components in the sensor head. And it's going to ensure that your leaf is at something that's close to ambient temperature, right? And that's going to change throughout the day. And so this is something that you have to reevaluate in the context of what time of day you're making the measurements. Okay. I'm going to pause here and adjust my leaf because it's not responding. Let's try. We'll see where that gets us. I picked a different leaf and I turned the light intensity down. Maybe it was just too much for this plant that's been setting in the dark. Okay, so let's talk about response measurements now. We have sort of two really common response measurements that are made with the instrument, though there are a number of other different kinds of responses that we could measure, right? So the two most common ones are response to carbon dioxide concentration, what we call an ACI curve, and response to light, which for leaf level measurements we generally refer to as an AQ curve. The other kind of measurements we could make with the instrument for response is we can make measurements where we look at response to temperature or response to VPD, or depending on the light source that we're using, the response to changes in spectral distribution by changing how we control the LEDs, right? But ACI and AQ curves are by far the most common, and the interpretation of those curves is very well established, such that we can derive known parameters from them that tell us more about the physiology of the leaf than any point measurement by itself. So we'll start with a light response curve and a curve. So this is data that I collected on okra. And you can see I have 5PS2 plotted here in the blue, and I have the carbon assimilation rate plotted in the green. And what we can see is that okra is a beast. That's a very high carbon assimilation rate. And that's a very high 5PS2 for something that's approaching saturating light. This tells me that the plant can take a lot of light and it can do a lot of photochemistry with it, right? And we can see, as I talked about yesterday with 5PS2, that as we increase the light intensity, the efficiency at which the plant uses that light decreases, right? At higher light, plants are less efficient at using the light. Right? And that's because as we go to higher light, other photochemical processes start to become engaged. So if we take this curve out to even higher light, these things are generally of the shape of an exponential rise to some maximum. You can see I took this to something a little bit above full sun intensity, and we didn't quite reach the maximum. My guess from looking at the curve is that I probably would have had to go to 3000 ppfd before this thing fully saturated. From this curve, we can derive a number of very meaningful physiological parameters. So the first thing is that the y-intercept of the green curve is the dark respiration rate, right? So this is carbon assimilation in the dark, and it's why it's negative, because in the dark, the leaf loses carbon through respiration, just like we do. And as I said, this thing is an exponential rise to some maximum. That maximum value is what we call ASAT. It's the light saturated assimilation rate. And we can derive this parameter from measuring the entire curve, or once we know the light intensity at which this curve reaches its asymptote, we can make a measurement at that light intensity and say, that's ASAT. This is the point at which if we were to give the leaf more energy, it couldn't do anything with it. For leaves that are adapted to lower light than something like okra, if you continue to go to higher and higher light intensity, that maximum value actually starts to go down, right? You see photoinhibition or photo damage, but that's difficult to do in typical C3 crop plants like this guy. Okay. The point at which the green curve crosses the x-axis, we call the light compensation point. And this is the value where the carbon assimilation rate is 0. Or you could say it's the point where respiration is balanced by assimilation, right? Such that the leaf is losing exactly as much carbon as it's taking up. When we fit a straight line to the initial portion of this curve, right? The slope of that line we call the quantum yield, or the apparent quantum yield of carbon assimilation. And it's like what I showed yesterday when I was showing my fluorescence data, and I said, well, in this experiment with tobacco at low oxygen, we measured a slope of 8 here, right? That was 8 photons per carbon, right? Plotted the way this is plotted, that'd be a slope of 0.125, right? That's the theoretical minimum for a C3 plant. I don't know what the slope is on this one. I didn't fit it, but it's drawn on here for illustrative purposes. Okay. So the way we do these measurements, we're going to use fixed sample CO2 control, right? So this is a different example light response curve. And I have delta CO2 drawn on the curve here. Right? And you can see at the saturating light intensity, this delta was 45 ppm. And at dark, it was -1 ppm, right? So just like for my survey measurement example, if I were controlling reference at 400 here, well, this means at my highest light intensity, I would measure carbon assimilation at 365 ppm. And at the dark, I'd measure carbon assimilation at 401 ppm. It's hard to make a comparison between those kind of data points if I did that, because there is a photosynthetic response to CO2. If I control on sample, well, that means that when I measured this one, the sample concentration was 400, but the reference was 445.2. And when I measured this one, the sample concentration was 400, and the reference was -390, or was 399. Right? Very easy to make a comparison now. I don't have CO2 response nested within my light response. We use the same general guidelines for controlling water vapor, right? So we want to set it to a fixed BPD. And then one of the key things that we're going to do is we're going to set the instrument to control on a fixed leaf temperature. And so on a light response curve, as we're moving from one end of the curve to the other, we're changing the light intensity. It's the whole point of the curve. And at the same time, we're changing the radiated load on the leaf. And when we change the radiated load, well, we change how much the light is heating the leaf. So at this point, we have a light source that's providing a lot of heat to the leaf. And when we go to the dark, the light's not providing any heat to the leaf. The leaf's actually losing heat, right? So by controlling on leaf temperature, the instrument's going to take that temperature exchanger and it's going to make it warmer or cooler, whatever it needs to do to keep the leaf temperature constant as we move through the curve. That did not help. Well, okay. So we have two ways that we could make the light response measurement. We could start in the dark and we could increase the light intensity, or we could start at high light and we could decrease the light intensity, right? This data is from capsicum, bell pepper, where I did exactly that. I took the same leaf and I started up here at high light. I let it get perfectly acclimated so that I saw a constant assimilation rate. And then I just went down in light intensity. I went all the way to dark. And then I turned around and I came back. Right? And those curves look pretty much the same curve. Right? It would be hard to argue that they're significantly different from each other. Which tells me that if I do the measurement carefully, it doesn't matter which way I go. But there's a trade-off, like all things in life. So this is looking at sort of instantaneous data as I move between 2 points on those curves. So both of these, when we get out here, are at the same conditions. That's 250 ppfd. In the top panel, I start at 500 ppfd and I step the light down. And in the bottom panel, I started at 100 ppfd and I stepped the light up to 250, right? And you can see that I get the same assimilation rate, the same stomatal conductance, the same CI. right? Within as well as we can possibly measure them. And I know it looks like there still might be a little trend here, but that's just, it's within the noise, right? The way the data was captured. So what's the real difference here? We're getting to the same endpoint. We make a big step change down. We make a small step change up. Well, going down in light intensity, it takes, what, 2.5 minutes for the carbon assimilation rate to stabilize, right? At this point on, it's constant. It takes 6.5 minutes for the stomatal conductance to stabilize, right? At this point on, it was constant. When we go up in light intensity, we look at this, well, that carbon assimilation rate really isn't constant until we get to somewhere past the 15-minute mark. And we're at almost 20 minutes before the conductance is constant. Oh, okay. So this tells me that if I want to do the curve where I start in the dark and I move up in light intensity, it's going to take a really long time between each one of those data points. If I wanted to do 10 light intensities, and I wanted to wait for the conductance to stabilize at each one of those going up in light intensity, well, man, that might take me 200 minutes. It's a long time to get one curve. If I'm going down in light intensity and I want to do 10 data points, and I want to wait for conductance to stabilize, well, that might take me 60 minutes. If I don't want to wait for conductance to stabilize, well, then it might only take me 20 or 30 minutes to do the light response curve. This happens because, as I said, carbon assimilation changes pretty much instantaneously when we change light. So if we cut the energy into the system, the carbon assimilation rate drops near instantaneously. And in this top situation, right, there's a stomatal response to that carbon assimilation rate. So as we go down in light intensity, the assimilation rate drops. Our stomata were wide to support the previous assimilation rate, and now that the rate has dropped, they're going to slowly close because they don't have to let as much water out to get the same amount of carbon in to support assimilation. So in the top panel, the stomates are more open than they need to be, and we're really only waiting on the kinetics of the diffusion pathway, right? Assimilation changes quickly, stomata change slowly, but they're always more open than they need to be to support the new assimilation rate at the lower light intensity. When we go up in light intensity, right, the stomata were more closed than they needed to be to start with, right? So we put more energy into the system. The plant's going to try and ramp up the assimilation rate, but it's limited, right? It starts to deplete the intercellular airspace. The stomata sense this, they start to open up. And then the assimilation rate increases a little bit because we get a little bit more CO2 in there. And then the stomata sense that depletion and they start to open up. And so there's this feedback that happens as we go up in light intensity where the stomata are actually limiting carbon assimilation rate, not energy, right? So if the plant didn't have stomata, it would look just like going down. I mean, it would be a step change up in carbon, but they're there and they're limiting it. And the carbon assimilation rate can't stabilize until the stomata are close to whatever value they're going to be at for the new assimilation rate. Okay? So typically what we'll do when we do a light response curve is we'll take a leaf that's in an environment where it's already at a saturating light intensity, or we'll put it in a chamber at a saturating light intensity. We'll wait for it to stabilize. If the plant is happy, and it's been close to a saturating light intensity to start with, that might only take 4, 5, 10 minutes, something like that. If the plant has been in the dark, like my lemongrass here, it might take it 30, 45 minutes to come to steady state at that saturating light intensity. And then once it's at steady state, then we'll typically do the measurement where we go down in light intensity. There is a particular case where we would start in the dark and increase light intensity. And if we want to make a really careful characterization of the quantum yield, that initial linear slope, then we'll start in the dark and we'll slowly increase the light intensity. Okay? So on the Li6800, we have these things called auto programs and background programs. And auto programs are pre-built scripts, and I'll talk about what background programs are in a moment. But we have an auto program for light response. And this lets the instrument make the light response measurement, collect the data along the curve under instrument automation. So we can put the leaf in the chamber, we get the environment set up the way we want to get started. When the leaf is happy, we hit start, and then we come back later, and we have the light response curve. This tool is wonderful, right? It makes measurements very efficient. But before you use this tool to do your light response curve, what I generally recommend is that you go do one manually so that you can pick out what light intensities you want to make the measurement at. So you can pick out how long it takes to stabilize between those light intensities, right? That curve, it's a curve, right? What do we care about? Well, do we want to get the initial slope really well? Do we want to characterize the curvature? There's a parameter there called theta. I didn't really talk about that. Are we interested in characterizing when the saturating light intensity is reached? Are we just interested in the max and the min values, right? That answering that question tells us how many points we want and where we want to put them on the curve, right? So when I do my first light response curve on a new plant, I will set the light intensity to some mid-range value. So if I think the plant might saturate at 2000, I'll set it to 1000 or 1200. And I'll work my way up in light intensity until it's nice and flat. And then I'll turn around and work my way down. And then once I've done that, I'll pick what points I want. And then I'll put them as my set points for this QN value. And I'll use those to do my light response measurements under instrument automation. These automated curves are really wonderful because we have this thing on the instrument called stability criteria. And stability criteria allow you to make a statistical definition of what good data looks like, right? And we have two kinds of stability criteria on the 6800. We have user stability values, and then we have environmental stability values. You can see these little green icons here, and it says 5 of 5 or 3 of 3. This means for the environment that we have 5 environmental controls operating, and the instrument is controlling all of them at the set point that we want them, right? If it wasn't, this would be yellow, and it would say 3 of 5 or 4 of 5, depending on how many weren't functioning the way they're supposed to at that point in time, right? The instrument checks these automatically. You set up user stability, typically based on biological variables, and the instrument will watch these to decide whether or not it should log data or if it should wait longer. And the values that I look at when I'm doing a light response measurement with fluorescence is I look at stomatal conductance, care about that. I look at this parameter we call F, that's the raw fluorescence signal. That is a direct probe of the physiological state. If the fluorescence is changing, the physiology is changing. If the fluorescence is steady, then all of the physiology associated with photosystem 2 is at steady state. And then I look at carbon assimilation A, right? I don't use the Li-Cor defaults, right? You can see right here, this default, oh, these are probably JSON setup values, but all of these defaults are 1 to start with. And if I started with a default of 1 and I characterized it and decided 0.005 was the right value, the instrument's gonna think that it's really stable if I'm using that 1, even though it's not stable at all because the real threshold is 0.005. My leaf has started to behave, so we're gonna get to do a real experiment. And part of doing that real experiment is I will show you how I set these stability criteria. So we're gonna do that, okay? Don't use our defaults. Once you have these characterized the way that you want them, the instrument's gonna remember them, but you can also save them as a configuration. I should also say that for people that work in a lab where there's a shared instrument, or that instrument is being used by multiple people to go collect the same data in the field, that you can create a stability criteria for anything. And so if you were doing survey measurements and you wanted everybody to log the data when it reached the same level of stability, you could set one of these definitions, and then all the people that are out collecting the data could look at this and they could say, oh, it's 3 of 3, I can log a data point now. Right? When we do a response measurement, the instrument's going to look at them for us. But again, we're going to characterize these. We're not going to just use the defaults. The way that we characterize them is we put a leaf in the chamber the way we want to make the measurement. We wait for the graph to become a nice flat line. It's not perfectly flat, like there's noise, right? Some of that's feedback from the environmental controls, some of it's just noise in the sensors. So there's a little wiggle to it. And what the instrument's going to do is for this standard deviation or this slope limit, what it's doing is it's got a box of data here. In this case, it's using 15 seconds, and it's moving that box one data point at a time, that 15-second window, along this curve. And it's computing the slope and the standard deviation as it goes. Right? And so because there's some noise to the data, that slope and that standard deviation are going to change a little bit. But when my data, the mean value, so the nominal slope is 0, it's not going to go above some value. Whatever that value is that it doesn't go above, that's what I use as my threshold, right? Because I know that when this value is staying below whatever number I've picked here, the line on my graph looks like this. And if it's bigger than that, the line doesn't look like that. We'll do this. I'll do it with my instrument so you can see how I pick it. Okay? When we do a light response curve, we're going to let the instrument decide whether or not it needs to match for us. This is a nice feature because then we don't have to think about it. We're going to use match if, typically, and the instrument's going to check these criteria that are a lot like the criteria that I gave you yesterday for when to match. And if any one of these criteria are met, then it's going to go ahead and match the analyzers. If none of them are met, then it says, I don't need to match before this data point, and it'll save you the time that it takes to do the match. Generally, the way a response curve works is that if we were looking at the data in real time, that's the blue strip chart here, is that we're going to log a data point when things are nice and stable at the previous set point, and then things in the chamber are going to change, right? And it's going to change because we changed the chamber environment, and so the physiology is going to change. And the instrument's going to compensate from that, and things are going to move around. It might not be a nice, beautiful curve like this, but it's going to do something. We don't care about what it's doing during this time. So when we set up the response curve, we define a minimum wait time. And that is this period of time where we're just ignoring everything that's happening. So in a light response curve, I typically set this relatively short, maybe 45 seconds, maybe 60 seconds. I know that carbon assimilation is certainly not going to be stable in that time frame. Conductance is definitely not going to be stable. So I just ignore that first minute. That's my minimum wait time. And then once that minimum wait time is reached, the instrument's going to start looking at those stability criteria that we've defined, right? In the 6800, we're going to use early matching. And so it has a second set of stability criteria just for the reference analyzer that are hard-coded in the instrument. And if we're allowing for early matching, when the instrument decides that the reference line is stable, irrespective of what the chamber is doing, it'll go ahead and match the analyzers. That's what's happening right here. It's going to come out of match mode, and it's going to continue to check the stability criteria. And when the stability criteria are finally met, or we've reached what we call the maximum wait time, then it's going to log a data point and move on to the next step in the curve, right? So if we happen to set our stability criteria too tight, we didn't do a good job characterizing them, or maybe something weird happened with that measurement, this maximum wait time is just the timeout. So we're going to pick a value for this that's longer than however long we think it typically takes things to stabilize. So if I'm doing a light response curve, I know it takes 5 or 6 minutes for conductance to stabilize. And so I might set my maximum wait time to say 6 minutes, because I know by that point, carbon assimilation is certainly stable, conductance is probably stable, and if I haven't met my stability criteria, I probably did something wrong, and we'll just keep going. Okay? So let's talk about the ACI response. It looks very similar to a light response curve. It is, however, not an exponential rise to a maximum. Here we have the carbon assimilation rate plotted versus the CO2 concentration inside the leaf. Remember, that's the derived parameter that we get from the fixed law formulation. I don't remember what this data is from, but it's from a C3 plant, maybe soybean. We use the ACI response first and foremost to fit for the enzymatic efficiencies of photochemistry. And so in a C3 plant, the ACI response is well characterized, and it's fit for 3 different regions. So this initial region of the curve is fit for a model that describes a parameter we call BC max. This is the maximum rate of carboxylation by Rubisco. In this region of the curve, the plant is carbon limited, right? As we increase carbon, we see a concurrent increase in the assimilation rate. This next region of the curve, with my green dashed line drawn on it, up here, we fit for a parameter called Jmax. And Jmax is the maximum rate of electron transport mediated regeneration of RUBP. It's a whole big mouthful. But it basically is how fast we can get electrons down the electron transport chain. Once we reach this point in the curve, we have more carbon inside the leaf than the plant can deal with. And so the limitation is how fast it can get electrons down the electron transport chain to produce the energy that we need to reset RUBP so that we can fix the next carbon. So we're electron transport limited. It's important to note that this portion of the curve is not an asymptotic portion, right? This is not a flat line. As we continue to increase the CO2 concentration, despite the fact that we've reached our maximum electron transport rate, there is an increase in the assimilation rate. And that increase is happening because as we continue to increase the CO2 concentration, the oxygen concentration is remaining constant. And so we're changing the CO2 to oxygen ratio such that fixation of oxygen by Rubisco becomes progressively less and less and less common, right? So by increasing the CO2 inside the leaf, we're actually depressing photorespiration. And so for the same electron transport rate, we actually fix a little bit more carbon. That's why this continues to climb. It's why it doesn't reach a maximum value. I didn't take this data set all the way out to do this, but if you continue out to high CO2, you reach a different portion of the curve that's fit with this gray model for a parameter that we call VTPU, or you sometimes just see it called TPU. It's called triose phosphate utilization. And the limitation there is in the regeneration of triose phosphate, which is limited to the phosphate, or related to the phosphate status of the plant. Right? Again, this is in a C3 plant. We can do this same measurement in a C4 plant, but the interpretation there is a little bit different, right? Because Rubisco is not the first enzyme in the carbon fixation process, right? It's being fixed by PEP carboxylase or some group of related enzymes to make a C4 organic acid before it's decarboxylated elsewhere and then refixed by Rubisco to make our sugars that we're interested in. We can get other parameters from a CO2 response curve. So just like our light response curve, the point where this curve crosses the x-axis, right, assimilation of 0, is called the CO2 compensation point. we call it gamma star. Right? This is the point where respiration is balanced by carbon assimilation. This point changes with photosynthetic metabolism, right? The CO2 compensation point and the initial slope of this curve is different whether we're a C4 plant or a C3 plant. And so these curves are often used to screen plants of unknown photosynthetic metabolism to see what they're doing, right? It gives us a good first approximation. I'll give a reference later that describes this in more detail, but there's this parameter called stomatal limitation, and it's fit as sort of a demand function between the carbon assimilation rate observed when the CO2 concentration in the airspace of the chamber is at ambient, and so the CI value is something less than ambient, and the carbon assimilation rate measured when the CI inside the leaf is the same as the nominal ambient value, right? So if the nominal ambient value is 400, well, when I measure assimilation with 400 at the CO2 concentration in the chamber, I get this assimilation rate. When I increase the CO2 in the chamber, such that the inside of the leaf is now at 400, I get this assimilation rate. The difference between these two parameters is how stomata are limiting photochemistry, right? Because if the stomata weren't there, there would be no difference between these numbers, right? So stomatal limitation is something that changes with water stress, right? Because that changes stomatal aperture. And yeah. So the way we do these curves is we always use reference CO2 control. Look at that, it's completely different than everything I've told you thus far. So we use reference CO2 control here because it's the fastest control mechanism, and because we don't care what actual CO2 concentration we're at, right? We're gonna make a curve that's a function of CO2 concentration. And so we can just apply the appropriate models to fill in the differences. We don't care where the actual data points are. So we go for the fast method. I'll explain why we want to be fast in the next slide, I think. We do this measurement at a saturating light intensity. We don't want to confound the CO2 response by being in a condition where light energy is limiting to photosynthesis. Right? So to know what a saturating light intensity is, we don't just look at a plant and pick a number. We don't flip through the literature and use what our colleague used. We have a photosynthesis system. We go measure it. This implies that before you do CO2 response measurements, you should probably do a few light response curves to figure out what an appropriate saturating light intensity is. Because while there are certain physiological limitations, physiological constraints that vary by species, genotype, cultivar, right? There's physical bounds to it. There's also an environmental component to what the saturating light intensity is, right? How the plant responds to the environment. If I take the same plant and I grow it in a growth cabinet at 100 or 200 ppfd, and then I take that same plant and I grow a clone of it in the field under full sunlight, and I go measure light response on those two, the one grown in the growth cabinet will saturate at a much lower light intensity than the one grown in the field, despite the fact that they could be genetically identical to each other. So we want to characterize it. We don't want to just use a number. We make the same rule for humidity here, and we control leaf temperature when we do a CO2 response measurement. And we have two choices in that leaf temperature control. We can either control at 25 degrees C, or we can control at whatever the current value is. And when we go back and process our data to fit it for those different regions for VC max and J max and TPU, then we can apply the temperature correction to adjust our curve to 25 degrees C, because all the enzymatic models for that are based on 25 degrees C. So the way we do these curves, here, there is absolutely a right way and absolutely a wrong way. So what we want to do is we want to acclimate the leaf at nominal ambient conditions for CO2, so say 400 ppm, and a saturating light intensity. And then we want to go down in the CO2 concentration very quickly, and we want to immediately return back to the nominal ambient value. We typically repeat a few data points at the nominal ambient value. So you can see there's a group of them clustered here. One of these is my first data point before I went down. 2 of them are my data points after I returned. And then we're going to go up in CO2 concentration. And we do the curve like this because when we go to low CO2 and we start to deprive the leaf of carbon dioxide, what can actually happen is we'll start to turn off rubisco. So rubisco has to be physically bound to a CO2 molecule to be functional in the first place. When we start stripping those away, it fixes it, it's done. When we go to high CO2, there's a stomatal response to CO2. And as we start to move up in the CO2 concentration, what's going to happen is the stomates are going to want to start to close. This isn't such a big deal, but you can see the functional effect of it here. So the blue data came from a 6400, which isn't very fast to go up in CO2 concentration compared to a 6800, which is very fast. And it took it quite a while at the high CO2, right? Minutes. And the result is that as it spends longer and longer at the high CO2, the stomata start to close, and that starts to limit the achievable CI that I can get to. The curve is still more or less the same, but I can't really fit it out here because I can't get that high CI concentration, right? So that's a good argument for why we want to do these curves fast. The other thing that we want to think about is what kind of metabolism we're making these measurements on. If we're using a C3 plant, we never want to go to 0 CO2. If we do that, we will absolutely deactivate rubisco. If we're doing a C4 plant, right, there's a different enzyme between the atmosphere and rubisco. We always take a C4 plant to 0. Just like a light response curve, there's a pre-built auto program on the instrument to do the CO2 response, right? This is called CO2 response. We can pick our CO2 control. We can pick the values that we want to make measurements at. You can see here, this was set up to do a C3 plant. So we start at ambient, we go down to 50, you might go to 30, 20, somewhere in there. And then we turn around, we repeat data points at ambient, and then we start going up in concentration. We generally want to do these curves quick. You can see I have 60 to 150 seconds between data points. That's pretty reasonable. The one difference here is that unlike a light response curve, we want to do this curve fast, and the instrument is always faster than I am. So when I'm approaching new plant material for CO2 response, I start with the auto program. I put in a series of values that seem reasonable like this, and I go make the measurement, and I see how the data looks. And then if it's not good, then I change my values, I modify my curve, I do it again. And so I might do a few different CO2 response measurements on a couple of different leaves in slightly different ways to come up with the consensus for how I want to make this measurement in the first place. And then once I've done that, then I'll go start collecting the real data for my experiment. So here, because we're changing the CO2 concentration between every point that we're making measurements at, we want to make a match adjustment at each one of those points. The traditional way to do this is to use what we call always match. And this just forces the instrument to match the ergos before it logs each data point. In the latest version of the 6800 software, I showed this yesterday, we have this thing called range matching, where we characterize the match offset as a function of concentration. And so we can, for CO2 response using 6800s with the software, what we can do is, before we collect the curve, we can do a range match. It takes 3 or 4 minutes. And then we can go make this measurement. And if we happen to be making this measurement in the lab, or someplace where the temperature is reasonably stable throughout the day, then we might be able to do one range match at the beginning of the day, and that applies for all of our measurements through the whole day. And by using range matching, we save a lot of time. If we're going to use always matching, then we need to set always match in our match options under log files. So that's how we do a steady state CO2 response curve, right? We've waited for the CO2 concentration to go and be steady at some value. We've waited for the assimilation rate to get there. We're going to give it a step change. We wait for things to stabilize at the next one, step change, stabilize. That's what we mean by steady state. The 6800 supports a different method for doing CO2 response curves for C3 plants. It doesn't work for C4 plants, definitely doesn't work for canned plants, right? But in C3 plants, we can use this methodology that's called RACiR, right? And this is work that came out of the University of New Mexico, Dave Hansen's lab. And this is a methodology for taking the instrument and using it to do a non-steady state CO2 response curve. So instead of waiting for the CO2 concentration to stabilize at any one value, and then the assimilation rate to stabilize at that value, what we do is we just continuously change the CO2 concentration. And we measure instantaneous assimilation concentration, right? This is data that I collected on tobacco using the two different methods. Let's see. I think I did this with two instruments that were head to head. And so they were clamped on different spots on the same leaf, and they measured it at the same point in time, right? And these are my steady state points in blue. And the green curve is the RACiR method, right? That is the same curve. They fall right on top of each other. And I shrunk the point size here so that you could see this a little bit. But one of the greatest things about this RACiR method is data density. So this blue curve, I have maybe a dozen of these data points. I think it took me about 45 minutes to make that CO2 response measurement. The green curve, these are individual data points. There's a little less than 300 of them on this curve. And the curve took 10 minutes. So it took a quarter of the time, and I got many times more data points. Now, when we think about what we do with a CO2 response curve, right, we're going to fit theoretical models to it and then get parameters from those theoretical models. Well, the fitting works a whole lot better if I'm fitting a few hundred data points down here to get BC max and maybe a few hundred data points up here to get J max, as opposed to fitting maybe 7 or 8 points down here and maybe 3 or 4 points up here, right? There's a lot more confidence in the curve fitting. So I like this method a lot. There's no magic auto program that does the RACiR method for you, unfortunately, right? The way that we think about doing these curves, we use the same environmental controls as the traditional ACI curve. One of the things that we do that is very counter to typical Li-Cor dogma is that we never match the ergos for RACiR, okay? This is really important. We don't care what the match offset is, because we make a measurement for RACiR with a leaf in the chamber, and then we make a measurement using the exact same experimental configuration on an empty chamber. And we use that empty chamber measurement to derive a correction that accounts for the flow-dependent time delay between when reference air and samples, or when reference and samples see the same parcel of air, right? So there's differences in this flow path based on the chamber volume and the length. And that means that when I bring air in from the console, it reaches the reference analyzer at a different time from when it reaches the sample analyzer. For normal measurements, this doesn't matter because we're at steady state. Here, we're transitioning. We're never at steady state. So this empty chamber correction implicitly contains the match offset and the flow delay, right? And if we're working someplace where the temperature is relatively stable, then we might do one of these empty chamber measurements at the beginning of the day, and then go do our RACiR measurements throughout the day. If we're working someplace where the temperature is not particularly stable, like out in the field, full sun, things are changing, then we might do one of these empty chamber measurements for every 3 or 4, maybe 5 or 6 RACiR measurements that we make, right? The way that we make these measurements is that we, just like our traditional curve, we're gonna acclimate at ambient, and then we're gonna step change to either our lowest CO2 concentration on the curve or our highest CO2 concentration on the curve. It doesn't matter which, but whatever way we do it, we want to make them all that way. And then we're going to provide a linear ramp in the CO2 concentration between those endpoints. In the paper, they use 100 ppm per minute. In practice, we find that depending on, I think primarily stomatal conductance, so what a typical stomatal conductance is for your plant material, you can go much faster. Something like sunflower that has conductances that approach or exceed 1 millimole per meter squared, 1 millimole per meter squared per second, you can go super fast. We've done the entire ACI curve on sunflower in 3 minutes, right? For things that have lower conductances, then we go a little bit slower with the CO2. We use never match. We use the auto program called Autolog. And Autolog just lets us log data at a fixed time interval. In this case, we're logging data every 2 seconds for 12 minutes. And I picked 12 minutes in this case because my entire ramp takes 10 minutes. So I will start Autolog a little bit before I start the ramp, and then I'll let it run a little bit after so that I capture the endpoints well. And then I use this thing called an auto control. These are under the environmental controls. An auto controls let us fit a function to a given environmental parameter. In this case, we're fitting a function, a linear function to CO2R. And that linear function says that we're going to ramp the CO2R from 10 ppm to 1,000 ppm over a period of 10 minutes, right? And when you hit start here, it'll start increasing the CO2 concentration. We don't want to start at 0 if we're going to start at the low end, because that'll turn the mixer off, and there's this stabilization period when it switches back on to go to that first 1 ppm value. So we always want to pick something that's a little bit above 0 for a minimum. From our empty chamber data, we're going to make a plot that looks like A empty versus CR empty, right? And we're going to fit that plot with a second order polynomial. In the original paper, they used a linear fit, and that works pretty well if you're only operating between the 0 and span values for the instrument. And it just happened that for the data that they were using, they were between 0 and span. But if you're operating over the full calibration range of the instrument, or something close to it, beyond where you've zeroed and spanned it, then you'll find that a second order polynomial gives you a better fit. And I'm sure that if you're paying attention, you notice that this data looks super noisy, but I want you to look at my y-axis here, right? This is a change of 1 assimilation unit. That's not noisy. So this fit gives us the actual assimilation rate as a function of, or an adjustment to the assimilation rate as a function of reference concentration. So we can factor out the residual from the match offset and the residual from the flow delay by recalculating based on the measured reference concentration in our real data, okay? I also like this data set because if you just factor this curve out of this data, take the residual here, it gives you the limit of resolution for how you made the measurement, which it's a pretty cool thing to be able to characterize. You have some estimate of uncertainty on the data. So I didn't talk about curve fitting curve fitting light response curves because they're pretty straightforward. In general, they're just fit as an exponential rise to a maximum. There's a number of different ways to approach it. There's a number of different models to do it. They're all empirical in nature. Some of them are based on theory, but for the most part, they're just empirical fits. And so it's pretty simple. You can take something like sigma plot, you can adjust your light response curve to factor out for respiration, and you can fit it to get initial slope and Pmax, and then take it back using their default model for an exponential rise to a maximum. ACI curves are not that way, right? The three regions that we're fitting are based on biochemical models, and things like sigma plot and Excel don't have those models as sort of native curve fitting applications, right? So we need something that is set up to do that. And Li-Cor doesn't make something for that, but there are a number of them in the literature. And so there's a few different ways to go about this, right? So this initial Long and Bernanke paper, this is a really great reference for stomatal limitation and how to derive that. They talk about uncertainties in gas exchange measurements. It's great. This Dubose paper gives a methodology of choosing the transition between VC max and J max, right? So when we look at this Sharkey paper, which is for many years the most common way that people fit ACI curves, because it's implemented in Microsoft Excel, it's very simple. So in this method, the Sharkey method, you have to pick that transition point yourself. You have to segregate the data. You have to say below this point is VC max, above this point is J max. This gives a mathematical method for doing it. Okay, the Sharkey paper has fallen out of favor, partly because of that reason, and partly because there's better tools to do it now. One of those better tools is run by Oak Ridge National Lab. It's called LeafWeb. And this is set up so that you can take your 6400 data or your 6800 data and you can upload it to their cloud-based platform, and they'll fit the data for you and return the parameters. A lot of people like that and they're happy with it, but it's kind of a black box. You don't really know the methodology they're using for the fitting. All you get is numbers back. And so in 2015, an R package was released. So R is an open source programming and statistical platform called Plant EcoPhys that's used to fit ACI curves. It does a number of other things, but it does the ACI curve fitting. It's completely open, so you can see what's doing in there. It's really easy to use. It takes 5 minutes to figure out. It's well documented in the original paper. And if you were to come to Li-Cor, you will find that we do the curve fitting one of two different ways. The vast majority of us just use this Dersma paper, the R package, Plant EcoPhys. And then there's a few holdouts that still use their own implementations. For the most part, we use this R package. And I would encourage you that if you're going to do ACI response, that this is a really great place, a really great resource to use. So I said I'd talk about auto programs and background programs in a little bit more detail. So functionally, they're the same thing, right? They're scripts that let the instrument do something under its own automation. Auto programs are pre-built scripts. You hit this little drop down here. There's a number of these in here. They have a little user interface where you can set things. In this case, I'm showing the generic response auto program where you can pick your choice of environmental parameter. In this case, it's the flash intensity. So this was set up to do that flash train experiment that I talked about yesterday in the fluorescence. And the instrument, you pick your environmental parameter, you give it min and max wait time, you give it stability criteria if you want, and it'll do that sequence, right? And you can pick any environmental parameter here, and you can pick subsets of environmental parameters. So you can say, measure different light intensities at different BPDs, right? And that's all pre-built. But sometimes there's things you want to do that the generic response or one of our pre-built auto programs just doesn't fit for. And then you can click this program builder button and it'll bring you to the interface for the background programs. And the background programs are just the GUI that's around a Python core that let you build your own program to make measurements. And you can set environmental controls, you can wait for things, you can manipulate variables in here, you can add user interfaces. And so when we want to do strange things at Li-Cor, Maybe I want to control my light intensity on an artificial diurnal cycle, and I want to make measurements at different times throughout that, and I want to do measurements of carbon dioxide and fluorescence during the day, and I only want to do measurements of the carbon dioxide flux at night, then I could build an auto program to tell the instrument to do that for me. And I would do that using this background program builder, right? And the background programs are neat because if there's some task that we want to have run in the background, we have another task that we want to make the instrument do, and then we want to do something else at the same time, they're not mutually exclusive. We can run several background programs at one time. So it gives you a lot of power and a lot of flexibility with what you can do with the instrument. If you think you have an application that needs one of these and you can't figure out how to get it set up, this is a spot where you should really call our support people, and they'll either tell you how to write it, or if you're really nice, they'll write it for you, right? Okay. So I have 2 more topics, and then we're going to take a little break, and it looks like we're actually going to be able to make a measurement. I changed to a different leaf, a different spot on the leaf, and it's responding. This is good. So the first topic, and this question was asked yesterday, and they said, when should I go make my measurements? And what I would say is that there's no one answer to that, right? It's in the context of your experiment and the kind of thing that you want to know. And so, you know, I said at the beginning, there's sort of this series of data that I think everybody should collect as they're starting an experiment. I said, okay, well, I gave a rationale for why you do a light response curve, right? It's good to know how a leaf responds to light. You should probably do some CO2 response curves. It's really useful to know how sensitive your leaf is to CO2, if nothing else, right? And then the other data set that I think everybody should collect is what's called a diurnal curve. And that's what's plotted on the screen here. And so this is carbon assimilation from survey measurements, where we just walked out in the field and we measured a leaf under the ambient conditions. And actually for each one of these, we measured a couple of leaves under the ambient conditions. And we did that from sunrise to sunset, okay, at various points in the day. And I have plotted here the carbon assimilation rate in blue and the light intensity, PAR, in green, right? Okay, so this isn't a perfect day. You can see some clouds came over in the late afternoon, but it's pretty darn close. On a perfectly clear day with no clouds, PAR is a nice bell-shaped curve like this. And I know that when I started making these measurements initially, and I see most people when they start making these measurements initially, they think, okay, well, just like Jason said many times throughout this presentation, light has the biggest impact on photochemistry. And so if light follows a nice bell-shaped curve throughout the day, then certainly photochemistry follows a nice bell-shaped curve throughout the day. And unfortunately, or fortunately, depending on how you look at it, that's not true. There are times in the day when photochemistry becomes decoupled from the light intensity. And you can see that happens in this data set. This is a perfect example of this. So in the morning, right, we come out of darkness, they start to track nicely, 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 and we start to reach peak light intensity. And somewhere before we get to the peak light intensity of the day, we reach the peak assimilation rate. And now the light's still increasing, but the assimilation rate is going down. And then we move past this sort of peak light intensity and the light starts to drop a little bit. And then all of a sudden the assimilation rate comes back up. And now we're doing more carbon assimilation at a much lower light intensity than we were a few hours before. And then eventually we hit some point in the afternoon where these things start to track each other again. So this period where assimilation has dropped in the day is called the midday depression, right? And this happens because as we start to approach the brightest part of the day, we're typically approaching the hottest part of the day. We're approaching the part of the day where VPD is the highest, right? So we're starting to put some water stress on the plant. Right? There's a response to the high temperature. There's a response to the VPD. As the plant has been doing photochemistry all morning, right, it's been building up starches inside the leaf, right? It has some limitation on how quickly it can get those out. As it starts to accumulate those starches, even without the temperature stress, without the VPD stress, it'll start to downregulate photochemistry. The other thing that could be happening here is that as we're shining more and more light on the leaf, we're actually damaging it, right? Every day leaves go through this cycle where light induces damage and they have to go repair that damage, right? So we're causing photoinhibition. And as we get up to these peak light, it just can't deal with it anymore, and there's this decline. Whatever the reason, plants show this, right? And this is really important thing. This is a really important thing. Because if I were to go in the field on this plant, and this data is from pak choy, a cabbage, right? And if I were to go out here in the morning, when things are nice and relaxed and happy, and I set my light source to a full saturating intensity and I do a light response curve, I'm going to get one curve that probably represents the true capacity of the leaf. And then if I were to go out in the middle of the day and I do that same measurement again, right, when I've seen this down regulation, well, I'm going to get a curve, but it's probably going to be a lot down here, right? Because I'm not measuring the true capacity of the leaf anymore. Like, there's some physiology going on that's preventing me from seeing what the true capacity is. I can't know how big this depression is or what part of the day it happens in until I go measure it. That's why these diurnal response curves are so valuable. Now, it's also really valuable in the context of the kind of experiment that I'm doing. Maybe I'm evaluating two different cultivars, and I'm looking for how they respond to some particular stressor. Well, it's quite likely that if there's a real difference in response, that they might behave exactly the same in the morning, but they might behave wildly differently during the midday depression. Right? In that case, this might be the best time to make the measurement. If I want to characterize something like a SAT, then maybe I need to be down here in the morning. Or if I want to do my CO2 response curve, I need to be down here in the morning. We can't know it until we make the measurement. Very valuable curve. So the next thing we need to think about is what leaf we're going to measure, right? Not all leaves are equal. There are, within a canopy, age gradients, light gradients, temperature gradients, turbulence gradients, humidity gradients. For any given leaf in that canopy at a point in time, they're not setting in the same light environment. They're not setting in the same temperature environment. They're not setting in the same humidity environment. They might be close. They might be relatively far apart. I'm doing something like a survey measurement where I'm trying to measure under the current conditions, and I'm just randomly grabbing leaves. Well, I don't know if the variation that I see is because these leaves are in different environments or because it's a real difference in physiology. So generally, we need to constrain for the leaf that we're going to measure. And so depending on the experiment we're doing and the kind of plant material that we're working with, that might be that we're choosing the first fully expanded leaf that's fully exposed to sunlight at X node positions back from the meristem, right? If we're working with something like corn, I showed that survey data set where we went really fast. I talked about going really fast, and I showed it stabilizing really quickly. In that experiment, we controlled for nodal position of the leaf, so where it is on the plant. We controlled for position on the leaf, right? Because corn leaves originate from a basal meristem, and so there's an age gradient along the leaf blade. So we wanted to measure at the same point on all of our leaves so that we were measuring leaves that were the same age. Right? And then we controlled for the orientation of that point relative to the sun. So there's a lot to think about here. There's not one right answer, right? but it is something that you want to consider and to control for as you do your experiments. Okay. That is my talk on environmental control. I think what I would say is that if there's a question at the moment about the presentation, that we can take a couple of minutes to answer a few of those. So if you want to submit them via the chat, somebody will tell me what's there and we can answer them. And then as you're submitting those, we can take a short break, maybe 5 minutes or so. And when we come back from the break, I'll answer those questions. And then we'll set up my instrument to do a light response measurement. Okay? And so go through sort of how we pick the stability criteria. We'll open a log file. We'll look at the graphs so that we can watch it collecting the data. And we'll get that measurement started. And then while it's making that measurement, I'm going to start talking through how the Li600 works. Okay? So submit questions. I'll stop sharing my screen for the moment. And then we can take a short break. And when we come back from the break, then I will answer questions.
Yes. Hello, Jason. Hello, Jason.
Hello?
Yes. Is this break for 15 minutes, maybe? 15 minutes.
Okay. 15 minutes? That's perfect.
We'll take a rest for you and we will continue. Oke. Yeah.
Okay.
Baiklah, Bapak-bapak dan Ibu-ibu, kita break dulu 15 menit, sekitar 15 menit. Nanti kita lanjutkan kembali. Terima kasih. Itu di belakang Pak Kukuh sama ini, Mas. Belinya sama lo tuh? Saya bukan sales. Yang install. Waktu itu tahun berapa tuh Pak? Mana? Yang di Unila? Kameranya buka. Oke, ya udah buka. Halo Jason.
Hello?
Are you ready?
I'm ready. Let's do it.
Okay. Yeah. For our next session. Okay. Your time.
Okay. I think there were a few questions in the chat. Do we want to go ahead and answer those now?
Yes. Okay. Okay.
I saw two. Let's see. So one of the questions from is asking about, let's see. So the question is, for the CO2 response curves that I showed, if I made those measurements under an artificial light or natural light, and those measurements were done using a light source on the instrument. So an artificial light, one of the LED light sources, just like the fluorometer that we see here. And we typically want to do them that way because, as the question asks, they talk about doing the measurements under sunlight, and when the sunlight changes, then the photosynthetic rate changes. So if we want to do a curve that takes a period of time and we need to constrain light, then it's best to use a light source that we control, because then the light is always constant. And when you look to the data that's published in the literature now, almost all of it is collected using an artificial light source for that very reason, because if you can't control the light, it'll add noise and uncertainty to the data. Okay. I thought it was two questions, but it may have been the same one. Okay, we're good. Let's look at, I'm going to start sharing my screen again. And we're going to look at the instrument. And we're going to set it up to do a light response measurement. And you can see that this leaf finally started to do something. You know, when we started and we first set it up, it was going maybe 0.5 for assimilation, and now we're at almost 8. I'm not really at steady state yet. And if I were making real measurements that I cared about, that I wanted to publish, that I was concerned with, I wouldn't start the measurements yet, because there's still quite a bit of slope here, right? The rate is still going up. And I know from measuring lemongrass before that this is a really low stomatal conductance. So it's outside of what's normal. And that's because it's 10 o'clock at night here. This plant has normally gone to bed a long time ago. So a more typical conductance for this plant, in my experience, would be somewhere around 0.2 under light saturated conditions. So we're way below that. And so it's not fully awake yet. But we're going to pretend like it is, and we're going to go ahead and do a measurement. I think we'll get a curve that looks reasonable, even though it might not be right. So I'm going to start this curve by just confirming that my environment is set up the way that I want it to be. So we walked through flow earlier. I don't have a particularly large delta. It's 6 ppm. So I think I'm going to actually turn the flow rate down to try and make that delta a little bit bigger. I don't have a whole lot of room to work here, but I'm going to set it to 300. Okay. I'm very happy with my VPD. I'm happy with the way that I'm controlling CO2. And I'm happy with the fan speed. But I'm going to go do my light response curve now, and I don't want to keep controlling the exchanger, right? This part of the instrument. Now what I want to do is I want to keep the leaf the same temperature in the chamber. And this is very easy to do once we're stable. If we just click T-leaf, now the instrument's controlling the leaf temperature at whatever the current value is. And so it was at 25.96, that I've just switched it to go at 25.96. We're going to change light intensity, so I'm not going to worry with that. I'm going to confirm that I like my fluorescence setup. So I'm pretty happy with these. I think this is okay. So we've already confirmed that we have the right leaf area in here. Let's go to the log files. And let's start setting up our logging options. So I want to record the data as an Excel file in addition to the text file, because the Excel file has all the equations built into it. I want to use additional averaging time. I like this, right? So instead of just capturing when things are stable, that point in time, and recording that to the data file, what the instrument's going to do now is it's going to take a 15-second window of data, and it's going to calculate the mean for that 15 seconds, and that's what gets written to the data file. So if my data is a little noisy because maybe I have a low photosynthetic rate, like we do in this case, because we just don't have much leaf area, it's not doing a whole lot, then the 15-second averaging will make the curve really nice and tight when we get down to the light compensation point. We can add extra data to the file if we want, but by default, all of the primary things are going to be in there. And I'm going to set my matching options. In this case, I'm doing light response, and so I want the instrument to think about whether or not it needs to match. I'm not going to worry with it. So I'm going to pick only match if, and I'll make sure the criteria are checked here, and only match if is selected here. When I do the log, I have an option of what I actually want to do with the fluorometer. So I could use the fluorometer, I could look at fluorescence, but I could not do an actual pulse of light, do the actual PAM measurement. Maybe there's cases where you would do that, but I want the PAM measurements tonight because I want to look at 5PS2 in addition to carbon assimilation. I probably want to look at electron transport rate too. And so if I pick nothing here, then it's not going to do the PAM measurement. And then if I pick one of these two options, it's either going to do F0, FM when it's in the dark, or FS and FM prime when it's in the light. And if I pick this option, it's going to do F0 prime as well. I don't have a need for F0 prime tonight. I'm not interested in QP or QL at the moment. And so I'm just going to ignore that measurement. And we're just going to do the normal PAM parameters. And then I have an option of what kind of flash I want to use, whether it's a rectangular flash or the multi-phase flash. And I know that my lemongrass likes light and that it's probably C4. And so I'm going to use the multi-phase technique just to make sure that I'm not underestimating my FM prime values. And now I'm going to open a log file. And I'm going to call this lemongrass. Oops. And I personally don't like white spaces in file names, so I don't use the space bar. but that's your own choice. And then today's date, which I guess is yesterday for the people watching me do this. So now that we have a file open, we're going to set an auto program up. Oh, and I don't like the scroll on there. We're going to pick light response, and we're going to choose our light intensities that we make measurements at. Now, you'll note that the initial value here is these all values are set based on the last thing that I did this with. And this is not a very meaningful measurement setup. For the first thing, the first several values are all much higher than the light intensity that I've acclimated my leaf at. It doesn't make any sense to just jump to a higher light intensity and then automatically do the curve, right? I want to start where I acclimated things. And I know that this probably isn't a saturating light intensity for this plant, but it's 10:30 at night, and it's probably the best that it's going to respond to. So we're going to set a whole new set of parameters in here. I've spent all this time acclimating at 500. So I definitely want to start there. Hang on, the Zoom thing is interfering with my, there, that's better for the moment. Keypad. And then I'm going to work my way down in light intensity. And we'll do several data points here so that we get what hopefully looks like a nice curve. And I'm just arbitrarily picking values, increasing my data density as I go to lower light intensities. Okay, so that's where we're going to start. And now we're going to set some min and max wait times, and 30 seconds is much too quick for a light response curve. So I think we're going to pick something, let's give it 6 minutes on the high end. And we'll give it 2 minutes on the short end. And if the instrument needs to match, we'll certainly allow it to do that. And now, before I start this measurement, I want to characterize those stability criteria. And so we'll look back at my measurement screen here. Oh, let's zoom in a little bit. You know, you could almost make an argument that that's starting to round off. Seems like a bit of a stretch, but it hasn't been a ton of change over the last couple of minutes. So we'll go for it. The conductance is kind of noisy because we're at a really low value in the water control. But we're going to pick some stability criteria, pretending like those are nice flat lines. And so we'll just start with stomatal conductance. I think for all of these, I'm just going to look at the slope limit. Okay, and so here's the currently calculated slope. And it's 0, 0, 0 something. It's going to oscillate around. It got really tiny. There's 0, 0, 1. 1, 1. Okay, and it dropped back down. So I'm going to pick something a little bit bigger than the largest value that I saw there, which is going to be 0.00. Add it out a little bit and go 5. Okay. And then we're going to do the same thing for fluorescence. So the slope here is about less than 1, a little over 1, less than 1, a lot less than 1, about 1, 3 point something. So I think what I'm going to do here for the fluorescence is I'm going to leave this value just at 5. I mean, that seems pretty reasonable. Most of the values are below 3, but we'll pad it out a little bit. And then we're going to do the same thing for assimilation, where we're just going to watch this slope for a few minutes, and then we're going to pick a value that's bigger than whatever we see here. So 0.4 something, 0.4-ish, 0.3-ish, 0.4 something. It goes a little bit above 0.5, still above 0.5. So I think a pretty reasonable value here might be 0.65. So that's a little bit bigger than our maximum, a little padding, and I think we're good. Uh-oh, what happened there? Let's look at our measurements. I don't know what happened there. Hopefully it goes away. Let's see. Okay, so I think it was just some perturbation in the CO2 control. Maybe my soda lime is getting kind of old, and it's sitting down here with me talking and breathing next to it for hours on end. Okay. So we've set our stability criteria the way that we want them. We've got a log file open. We've set up an auto program to go ahead and make our measurements. It says this is going to take about an hour. And at the maximum, that's based on this time. And now we're ready to hit start. And the instrument's going to do the light response measurement. And while it does that, we'll talk about the 600. So I've hit start. We can see the progress bar starting down here. If we go back to measurements, we have the real-time graphs here that's showing us the strip chart of what's happening in real time. If we go to one of these other graphs, we can see now not longer a strip chart, but a XY plot where we have the carbon assimilation rate, the electron transport rate, and the light intensity in the chamber. And these have some sort of ridiculous default scaling because we haven't collected any data yet. But here's the current value, and here's the current value. This one's 0 because we haven't measured anything. And as the instrument starts to collect data, what it's going to do is it's going to rescale these axes so that it looks reasonable, and it's going to start to draw our light response curve on there for us. And so I'll leave this up, and periodically we can check into it. And then when it's done collecting the data, then we'll go ahead and make a measurement. And I think maybe it's already matched itself. Oh yeah, there it went. So it went into match mode and it automatically matched the ergas for it. And now it's coming back out. And what it's going to do is it's going to wait till these lines meet our stability criteria again, and then it's going to log a data point. And then it's going to change the light intensity. And maybe we'll wait because I don't think this is going to take very long. I bet by the time we get to the 2-minute mark, because we were already pretty much stable, that it's going to go ahead and do it. So our stability criteria, fluorescence looks good. It's only the gas exchange things, and that's because we just came out of match mode. Looks like carbon assimilation is probably there. Conductance is going to move around for a moment. Let's see what happens. So we've reached our stability criteria. And the moment that it decided it was stable, it logged a data point. The instrument paused for a moment because it was doing the flash. And so now when we go to G, we see that we have reasonable looking axes and we have some data points. So here's our first electron transfer rate, which says we're about 65-ish. And our first assimilation rate, which was right around 9. And so if we look here, we can see this QN value is the light intensity in the chamber. It's just dropped the light intensity in the chamber to 400. And it's gone into match mode. And when it comes out, it's going to wait for things to stabilize, and then it's going to log another data point. There might not be much change between these as I look at this, but we'll see. Okay. So while it's doing that, I'm going to pick a different share. And let's talk about the Li600. So I showed this momentarily earlier. This is the Li600. It's a new instrument. It is a rapid survey tool. We'll talk about what that means. And it's really an instrument that is complementary to the 6800. So yesterday, I showed a picture like this, and I had some stuff circled on here that I called gas exchange, and then I had chlorophyll fluorescence circled on here just like this. And I was using that primarily in the discussion of the 6800, right? And there I had circled carboxylation and then the CO2 flux here. And I said in the 6800, that's the primary thing that we measure. But we also measure water. That's how we get stomatal conductance. In the Li600, all we measure is water. It's a porometer, fluorometer. And so it measures the apparent transpiration rate. We use that to get conductance. That's our gas exchange measurement. And then it also includes a fluorometer that we use to measure the state of photosystem 2. So here you can see there's a small aperture where our leaf goes. And on the lower side of this is a gas exchange system that's used to measure the apparent transpiration rate and the conductance. That's the porometer part. And then you can see some optics here, these kind of glowy domes, right? That's part of an open chlorophyll fluorometer. And so I'll show this again, but these are the modulating light and flashlight. These are the optics that focus it here. And then in between them is a detector that captures those photons that get emitted off of there by fluorescence. The instrument includes an integrated PAR sensor. So unlike the Li190 on the 6800, it's not a separate sensor attached to it, but this cosine corrected sensor is built into the instrument, and it's always held parallel to the plane of the leaf, right? You can see the leaf clamp here. There's a small polymer gasket on the bottom side of the leaf clamp that we use to seal to the bottom of the leaf. There's an infrared thermocouple, an IRT, that views the entire leaf aperture, and that's what we use to measure leaf temperature. There's a flow meter. This instrument uses relative humidity sensors instead of infrared gas analyzers to measure water vapor. The sensors that we use are high precision, high speed sensors. It uses a micro pump to provide flow through the instrument. So this micro pump is actually based on a piezo element, which makes it very energy efficient and very tiny. We don't control the environment in the chamber here. Instead, we use a solid state buffer to regulate the water vapor concentration and keep it constant at ambient. I'll talk about how the measurement's made, but you can see there's a match valve here, which implies that this is also an open system, right? It includes an atmospheric pressure sensor. The batteries are integrated into the handle and they're charged through the USB port, and that's what we use for data transfer. And the instrument includes an integrated barcode scanner. So you can go and print barcodes that identify all of your plants or all of your different treatments. And as you go along to make measurements, you can take the instrument, scan the barcode, and it will integrate that data with the conductance and fluorescence measurements that it records. So data gets stored on the instrument in a database, not really in a file system. And so there's a computer application that we show here that when we plug the instrument USB cord into our computer, it gives us access to the data system on the instrument. So we download the data. It tells us about how many observations we have. We can pick those individual chunks of data and export them into summary files. It gives us access to configuration management. One of the really neat things about this instrument is that there's very little user interface on the instrument. There's essentially one button we use for measurements, and then there's 4 arrows that we use to navigate through, okay? And we don't need much interface here because the fluorometer is quite smart. What we can do is we can create a configuration that tells us what kind of data we want to record. It lets us build prompts that are used to annotate that data so that when we go and collect it, we see a list of possible prompt values here. Maybe plant 1, plant 2, treatment 1, drought treatment, light treatment, whatever you want to call it, and then you can pick it from that list to associate it with the measurement. And in the configuration, we can choose whether we want to use this instrument in what we call manual mode or auto mode. And in auto mode, it uses predefined stability criteria to decide whether or not it's stable and whether it should make a measurement for you, or if you need to wait a little bit longer. So the 600 is not like a typical porometer that's been in the market space for decades now. So the porometers that exist for measuring stomatal conductance are all based on what's essentially a closed transient architecture. So they have a leaf. Sometimes they measure both sides. For most of them, they only measure one side, just like we do. And to the bottom of the leaf, they'll press a volume. And in that volume, it's sealed, except for where it contacts the leaf. There'll be a relative humidity sensor. And so when you put that volume up against the leaf and you look at the relative humidity, it starts to climb, right? And this rate of increase is related to the transpiration rate in the volume. And so if you take the slope of some part of that line, it'll give you the transpiration rate at that moment in time, and you can compute conductance. But these kind of porometers have some real fundamental limitations to them. So one of them is that relative humidity sensors are based on adsorption, desorption of water vapor onto a solid state material, generally some like polymer capacitor material. And so that means that they have an inherent hysteresis to them. So the response of the sensor changes depending on which way the humidity is going, right? The sensors also tend to be highly nonlinear. So there'll be a region in the middle where the response is sort of one-to-one. A change in humidity is a one-to-one response in the sensor. But as you start to approach the endpoints, that response starts to flatten out. And so as you go to really dry conditions or really wet conditions, the response of the sensor attenuates. In fact, most of these relative humidity sensors start to saturate somewhere before the actual saturation dew point. And their change in response as they start to approach that becomes very tiny. For some of these typical porometers, you know, one of the things that you have to do with them is after you make a measurement and the humidity is built up in them, you have to open it up and shake it to flush it out with ambient air to be able to bring the sensor back into a region where it'll actually read correctly. The 600 doesn't work that way. The 600 is a steady state porometer. We use the same basic approach we use in the 6800. It's a tiny serial flow path. So here's our microblower, here's the solid state buffer, The air goes to a flow meter. It gets measured by the reference relative humidity sensor. It interacts with the leaf, and then we measure the sample relative humidity. The solid state buffer keeps the water vapor concentration going through the instrument constant at some ambient value. So if you happen to be breathing around the instrument and contributing water vapor, or a big gust of wind comes up and it's drier than the air around you, those oscillations don't make it into the measurement, right? That's how we get away with not actively controlling it. That means also that our measurements are always made at near ambient humidity, which means that for our relative humidity sensors, they're always operating in the range in which they perform the best, right? We're never approaching saturation. We're never approaching completely dry. I talked yesterday about mass balance in an open system. And in case somebody wasn't with us then, I'll go ahead and reiterate it here. But basically, just like in the flow path I showed, we have a constant flow of air entering and exiting this volume that's oppressed to the bottom side of the leaf. And that air enters at some water vapor concentration that we're measuring with a relative humidity sensor. And then using that relative humidity sensor and the pressure and temperature, we're calculating the water vapor mole fraction. And that air interacts with the leaf, and the leaf transpires E, water vapor, into that air. And the air leaves at a different mole fraction and a different water vapor concentration. And we use the same mass balance approach that we use in a 6800, where we take what goes in and what goes out, that difference times the flow rate in, divide out by leaf area, which is fixed based on this aperture size, and that gives us the transpiration rate. And then from that, we make the same fixed law approximation, where we take the measured transpiration rate in the porometer, and we solve for total conductance based on the humidity in the chamber and the leaf temperature. And in this case, it's probably hard to see, but like I said, there's a small infrared thermometer that's being used to measure the leaf temperature. And that leaf temperature measurement is integrated over the whole leaf area in the chamber. That gets us total conductance. And then we partition out the boundary layer conductance, because just like in our regular 6800 measurement, there's turbulence in here that's making the boundary layer thinner, but it's not zero. And then we solve for stomatal conductance. It's very analogous, but then very different, because we're using different sensor technologies and a different flow path. So it's tempting to take the two instruments and try to make direct comparisons in their conductance measurements. And there are ways of doing that, but you need to keep two things in mind if you're going to take these out and use the data together. So one of them is that the 600 is a single-sided measurement. We're only measuring the bottom of the leaf for stomatal conductance. The 6800 is a two-sided measurement, right? We're measuring the top of the leaf and the bottom of the leaf to get our conductance. But in both instruments, we're still standardizing to the projected leaf area, right? Now, how different a one-sided and a two-sided measurement are for a conductance value are a function of how stomata are distributed on the leaf surfaces. If I'm measuring a leaf where all the stomata are on the bottom of the leaf, then my 6800 and my 600 will give me the same conductance measurement. If I'm measuring a leaf where the stomata are evenly distributed on the top and the bottom, half on the top, half on the bottom, then the value that I get from the 600 should be about half of the value that I get from the 6800, right? Because I have half the stomata in here as composed to there. And the other thing that we want to think about when we're integrating data between the two is that stomata distribution may not be uniform across the leaf surface. And so we have to think about spatial averaging. In the Li600, we're measuring a tiny little leaf area, 0.75 centimeters squared. In the 6800, we're measuring somewhere between 2 and 36 square centimeters of leaf area. And so if we have a leaf that's very heterogeneous in the way the stomata are distributed, then the way that we deal with that here is we make multiple measurements on the leaf. So let's talk a little bit about the fluorometer in here. We won't spend long on this, but as I said, it's an open fluorometer, right? So the sun serves as our actinic light source. We have optics and LEDs that provide the modulating light to get the wiggle, and that provide the saturating pulse, the pulse part of the PAM measurement. And those LEDs are focused on where the leaf sets right here. The detector for fluorescence sets in the middle, and then this is filtered so that it only sees a range of wavelengths that are emitted by leaves for chlorophyll fluorescence. Because the sun serves as our actinic light source, we make a light adapted or dark adapted measurement depending on whether we're outside in the dark or we're outside in the light. So if we go out at night or pre-dawn before the sun comes up, we can make dark adapted measurements and get things like FB over FM from the instrument. If we go out in the middle of the day, as shown here, when the sun is up and shining light on our leaves, then we're making a light adapted measurement and we're measuring 5PS2 or ETR. The fluorometer that we use in here uses the same basic technology that we have in the 6800, and it supports some of the same protocols, right? The main thing being whether you're using a rectangular flash or the multi-phase flash to estimate FM prime at infinite light intensity. And we talked yesterday in detail about how you would choose the difference between those or what your flash intensity would be. So what might we use this data for? Well, stomatal conductance and 5PS2 ETR, or FB over FM, that's a subset of the data that the Li6800 collects, right? So by its nature, the Li600 provides data that's complementary to the data that we collect with the 6800. The parameters that we measure here tend to be sensitive stress indicators, right? Conductance is generally a sensitive stress indicator of water status, right? So as plants become water stressed, stomata tend to close. 5PS2 is a sensitive indicator of any sort of stress that inhibits the plant's ability to capture light and convert it to do photochemical work. And so generally, these are stressors that degrade the photosynthetic machinery. So this could be exposure to excess light that damages the leaf. This could be accumulation of short-term stresses related to temperature or light throughout the day, or long-term stresses related to temperature over some developmental period. And that could be high temperature stress, or it could be low temperature stress. And so I have some data that I've collected here. And one of these data sets will point out, we'll talk a little bit about why it's so difficult to make measurements using ambient light outside for some of these things that show sort of the complementary nature between different instruments. And so the first data set's just an example diurnal curve. It is not a very good one because unfortunately, the time that I had to go collect this data happened to be a particularly cloudy time in Nebraska. And so this data is from one of the North American native hibiscus species. It's actually the plant that was originally used to flavor marshmallows. Anyway, so I have some of these growing. I set up an Li190 to collect ambient light, and I set it up at around sunrise. I took it down at about sunset. And so it's just recording instantaneous light throughout the day. And you can see we have more or less the bell-shaped curve if we just take the peaks here. But unfortunately, I had clouds passing over, so you can see these frequent drops in light intensity. And that's contributing to some of the variability in these variables. But essentially what I did was I went and measured 5PS2 and stomatal conductance throughout the day. You can see 5PS2 up here, When light is low, it's approaching our theoretical maximum, which is FV over FM. Oops. And then as the light picks up and the plants start to see it, they start to do more photochemistry, their efficiency decreases, conductance starts to increase to support that, 5PS2 drops. We reach the midday, and then you can see that there might be some midday depression here in conductance. It's hard to tell because it's a bit noisy. But it's a diurnal curve done with the 600 across one day. So the point of this data set is not really to show you another diurnal curve. At the same time that I made that data collection, I took my 600 or my 6800 and I clamped it on one single leaf of this hibiscus plant. And I made instantaneous measurements with it of stomatal conductance and regular measurements of 5PS2, right? And I set it up to control the chamber at the same light intensity that 190R was giving me. And then I took my 600 and I surveyed a whole bunch of leaves, each one of these. Well, I say a whole bunch. It's 5 leaves at each one of these data points that were all leaves that looked like the same as the one the 6800 was on. And the point of this data is to say that measuring one leaf in a canopy, as you can see here, right? It's sort of the same trend, right? Conductance rises, conductance drops off. 5PS2 drops off, 5PS2 rises as light intensity goes back down in the afternoon. My mean values behave roughly the same, but they're not the same value, right? It's the same trend. Simply measuring one leaf under the current conditions doesn't tell you what a canopy is doing. It tells you what that one leaf is doing. If you want to know what a canopy is doing at that point in time, then you need to make several repeated measurements across a variety of leaves to characterize the canopy. And it's possible with either a 6800 or a 600, but it's kind of painful with a 6800, right? Even if I'm doing the best kind of survey measurement that I can, and I'm getting data in, say, 30 seconds, well, that means that maybe I'm only measuring 1 or 2 leaves a minute, right? Because it takes me time to get from one leaf to the next leaf. My 600's fast. So I just turned this on in auto mode. It's already matched itself. And now it's telling me that it's ready to clamp on a leaf. And so I don't think we're going to get a very good data point, physiologically speaking. But we'll just see how long it takes to make a measurement right here. We'll see if it seals. This thing has a really thick midrib. So it's searching for stability. And there we went. It decided it was stable and it was ready to log a data point. And it did that in what, 5 seconds? So typical measurements from a 600 are somewhere between 5 and 15 seconds. It's a very fast instrument, which makes possible really large sample sizes when you want to go do survey measurements. Where I see the 600 being absolutely incredibly valuable is if you think about like a large-scale field experiment where we have a number of different varieties or genotypes that we want to screen for. And we have a whole bunch of these setting out there. And we want to find the ones that are physiologically interesting. Maybe this thing's under a massive drought and we want to find the couple of ones that still are maintaining high conductance or high photosynthetic efficiencies during this. Well, if I can make a 5-second measurement, I can screen a wide number of plants. I can have replicate measurements on each one of those plants, and I can pretty quickly find the ones that are interesting. Now, when we turn the story back the other way, right? We say one leaf under instantaneous conditions doesn't represent what the canopy's doing. What we can say that's true in the opposite is that if we choose a representative leaf and we make a highly constrained measurement, something like a light response measurement, well, that light response measurement is representative of how the other leaves in the canopy behave, right? So this data is from okra. And the solid data points, again, we're looking at phi PS2 in the top. We're looking at electron transport rate in the bottom. So the solid data points I collected with an Li6800 on one leaf. I did the same experiment we're doing with the lemongrass right here. So I got it acclimated at a saturating light intensity, and I did the light response measurement, right? And then I took my 600, and at the same time I was making the light response measurement on the one leaf, I went across my ochre canopy, and I measured leaves that were in different light environments, right? So some of those leaves are shaded, some of those leaves are in full sun, some are tilted away from the sun, some are tilted towards it. And the Li600 data are the open circles. And when I plot those open circle measurements with the solid symbols from the 6800 as a function of light intensity, these survey data points where I'm characterizing the canopy under the current conditions are representative of what the one leaf does when it responds to that same variable that's varying through my canopy, right? So when I find a physiologically interesting plant to work with in that large scale screening scenario, right, I can take my 6800 and I can apply it to make these highly constrained physiological measurements. What we've talked about today, light response, CO2 response. And I know that this plant is interesting. I found out that it was interesting really quickly with my 600. And then I can take that highly constrained data and I can characterize it. And I know because when I make these constrained measurements that one leaf might be representative quite well of how the broader plant behaves, if I've done a good job picking that leaf, then I don't have to make as many of these constrained measurements, right? So I can find the plants that are interesting, and I can get two different kinds of data that are complementary between the two instruments. So that's my discussion of the Li600. I'm going to go ahead and turn my instrument off here. And hopefully, at this point, the light response curve on the 6800 will be done or close to done. Oh, I think we finished it. I don't think it took a full hour. And I think we're doing pretty good. We're running a little bit ahead of schedule. That's excellent. So I'm going to stop sharing. Let's see. What do I do? So I stop sharing, and then I'm going to share again, and we're going to go back to the 6800. Okay, so we can see it logs some data points, and now we're at a light intensity of essentially 0. Let's go see how the curve looks. Well, it's a little noisy. I guess I'm not terribly surprised given how this thing is performing tonight. But for the most part, we look pretty good. We weren't entirely stable when we made our stability criteria, so that might be why we have a few of these points too. But if we were to ignore these two guys and fit that, that looks pretty much like a light response curve. And we can see sort of what we would expect here, which is that carbon assimilation decreases as we decrease light. That electron transport rate, which is supporting carbon assimilation, shows the same pattern. On, oh, that's versus CI. That's my raw fluorescence. Okay, let's plot some data and look at phi PS2. So I'm going to go back to log files. And I'm going to go ahead and close this log. And then I'm going to go to analyze logs. We're going to make some specifically interesting plots. This thing is, oh, where did it go? There we go. So there's two files that get created. So one is the ASCII text file and one is the Excel file. We're going to pick the ASCII text file because the instrument doesn't read Excel. And we're going to load it, and then we can look at things in it. And so if we go to data, all the data is in different groups. We'll get rid of constants, and we can look at gas exchange. And here are our assimilation values and our CI values. And if we scroll over, we can see stomatal conductance to water vapor, which is low. And then if we go to graph, we can make a plot. So let's do, on our horizontal axis, from the fluor light source, we're going to choose the light intensity. From our left axis, we're going to go to gas exchange, and we're going to choose assimilation. Here's our noisy data. It is what it is for 11 o'clock at night. And then from fluorescence, we're going to choose phi PS2. Should be in this list somewhere. And there's phi PS2. And you can see increasing in efficiency. And then it's approaching, oh, it's a little bit more than 0.6. I'm going to guess that this lemongrass might not be the healthiest lemongrass, or at least this leaf. So we can make a different plot here. So we'll get rid of that. Oh. Let's see. Oh, John, where's the clear it? Okay, so we're going to change, and let's look at oh, I'm doing this backwards. Let's go here and let's find gas exchange. Let's pick carbon assimilation. I'm just going to pick a different one there so it goes away. And then, no, not fluor light source. Fluor. We're going to see if ETR is linearly related to carbon assimilation over our light intensities, which it should be. Why do we only see 2 data points? $60? Nope. Where did they go? Oh, that's the issue. Let's fix the scaling here. 0, 60, maximum, yep. And with the exception of our couple of noisy points, that looks more or less like a straight line. Okay, so we collected some data. And now we need to get that data off of the instrument. And as we talked about yesterday, there's sort of 2 ways to do this, right? So that we can plug a USB drive into the back of the 6800, and we can use this dialog here at Manage Files to compile, and then we to the USB drive, and we can move them to our computer. The other option is that on a network, the 6800 will show up as a network hard drive, and you can just open it up in your file explorer and copy files off of the instrument that way. And I think at this point, we're running way ahead. I guess my hope was that the plant would do a little bit more than what it actually did, and that we could do a more meaningful light response curve, but we got something. And so I think now we can maybe just open it up to questions, and we can spend the rest of the time answering questions that you guys might have. So I'm here and available, and if you have something, go ahead and submit it, and we'll go from there.
Hello, Jason.
Hello.
Yes. Now the question session. Okay.
Okay.
Some question as you answered before, and now we add some question. The first is sorry. Okay. One question is this new Li-Cor, because integrated two equipment, how to cope with heat from both equipment?
Yeah, we actually, it's a really good question, because it's something that in the development process that we spent a lot of time thinking about and working with, right? Because when we do the fluorescence measurement, we're sending a lot of light to the leaf really quickly, and that heats it up. We take this measurement and we make this measurement outside under sort of full sun conditions, and so there's always light shining down on this. And so I can talk about a few ways in which we deal with this. So one of them is to keep the leaf from heating up when we make the measurement, just from sunlight shining on it and having it clamped in something, is that you'll note that this plastic is very opaque and it blocks a lot of IR. And what's hard to see is on the back side of it here, it's actually reflective. So there's a metal reflective material that helps keep the leaf from warming up here. And then you'll note that the instrument itself is vented. So there are water resistant vents that allow for free airflow around all the electrical components, which are basically built into a big heat exchanger. And then there's a fan inside here that's constantly circulating ambient air through it. And then our choice to make this instrument white is it's not an aesthetic consideration. It's because white materials like this reflect a lot of sunlight, and so they keep the instrument cool while it's out in the field. And so that's how we do it. So we have some active thermal management, we have some passive thermal management, but it's been really carefully designed so that it doesn't heat up and so that it doesn't heat the leaf. Okay.
Okay, sorry.
No problem.
Okay, problem, sorry. Okay, once more. With those frequent adjustments, does it mean that it is better to conduct measurement for same kind of leaf at the time? If the plant are on, say, polybag, will it better to measure them with bring them inside room?
Can you repeat the question? The audio cut out briefly, so I missed
Yes.
part of it.
Okay. This is from Unila. With those required adjustment, does it mean that it is better to conduct measurement for same kind of leaves at the time? If the plant are on, say, polybag, will it better to measure them with bring them inside room? Clear?
Yeah, I understand. So is it maybe better to measure the plant in the field or to bring the plant to some standard environment?
Yeah.
I think there's no right or wrong answer specifically. There might be a right or wrong answer depending on the kind of research that you're trying to do. It's not uncommon, particularly for people that work in greenhouse situations or in growth cabinet situations, where they'll take the plants and they'll put them under some artificial light source, not in the instrument, but maybe a halogen lamp or an LED lamp or something like that, to get them acclimated to a certain light intensity. And then they'll take it and put it in the 6800 to make the measurement at that light intensity. So the plant is already pre-acclimated. And that would be very common in those kind of situations if somebody was just interested in measuring ASAT or something like that. If you wanted a picture of what the plant is doing in the field, then you have to measure it in the field, because as soon as you bring it indoors or bring it into some artificial light environment, then you've changed the conditions the plant is seeing, and it changes its physiological rate. So it really depends on what you want to know. So no specific right or wrong answer. It's just really based on the experiment.
Okay, next question. Sorry, this is a silly question. It seems that you did the experiment ACI and CO2 curve using artificial light or indoor lamp. Is that right? Because the result so smooth. I had problem keeping the intensity of the light in the field constantly. Read the sun. And because the intensity of the light wobble, so the data become wobble. When I did the experiment, but I used different brand or IRGA, ADC, bioscientific, how to the light curve and keep the light in the field constant. Is it clear?
Yes.
Okay.
So for any sort of constrained measurement, like a light response curve or a CO2 response curve, we want to be able to control the light. It's really challenging to do these measurements under natural light. And so in the 6800, we like to use instruments that are chambers that include a light source. So like you can see here, I'm providing light, and that light is coming out of my fluorometer, and that light is at the value that I set it to. And it will stay there no matter what the sun does or what clouds do until I change it. And so, yeah, if you're gonna do a CO2 response curve, you know, those are properly done at a saturating light intensity. And that means that if you're gonna use the sun to do it, you're typically doing it at some time that approaches midday, because that's when light approaches saturating, though that might not actually be saturating for all leaves. And then you're gonna do it at constant light, which instantly becomes a problem if there's clouds or anything in the sky. So when you look at what's published for that data, almost all of it's done under an artificial light source. Yeah.
Okay, the next question. How to measure leaf respiration with Li6800? We wonder what technique to use. Clear?
Yeah, so yeah, respiration is seemingly simple and then very complex, right? So the simple approach to respiration is that you turn the light off so the chamber is completely in the dark, and you put the leaf in there, and you get a measurement of what we call dark respiration. And for a lot of things, it's good enough to assume that dark respiration is the always respiration rate. It's probably not, but depending on what you're doing, it might be good enough. If you want to measure respiration in the light, that gets more complicated. And that's done using a particular series of response measurements. It's not a measurement that I make often, so I don't know off the top of my head what the actual steps are to it. But I showed the commonly measured parameters document at the beginning of my talk tonight, and I gave a link to that document. And if you download that document, it talks about the different respiration parameters, whether it's dark respiration or respiration in the light, and it gives some references to a couple of papers by Owen Atkinson. And those give a methodology for measuring respiration in the light. And they talk about the difference between respiration in the light versus dark respiration. But fundamentally, it's something that the 6800 is perfectly suited to measure. And it's just the methodology to get the number. This is one of those things where I talk about how you apply the instrument changes the meaning of the value. Well, you're measuring carbon assimilation. But if you do it in the dark, the carbon's coming out, that's dark respiration.
With Li6800. It's clear? How to measure plant with small leaf with Li6800.
Yeah. So make this very dim. So for the plant that I was using tonight, you know, it has long leaves, but they're not very wide, right? It's a narrow-leafed grass. And if I clamp it in my chamber, right, it goes across the chamber, but it doesn't fill it completely. And in this example that I gave, it was pretty straightforward, right? I just measured the width of the leaf, and I showed you the leaf area calculator on the instrument to get the actual leaf area. Well, this is a pretty simple example because the leaf is more or less linear. For things with tiny leaves that aren't linear, well, then you have to know how much leaf area is in the chamber, but it's hard to see in there. And so in the fluorometer, there's a really nice feature. At the base of the fluorometer, there's a little spot where you can hook your fingernail, and the bottom comes out while the leaf is still clamped in the instrument. So now I can see the leaf in the chamber, and I can take my camera that's always with me, and I can take a picture of what's in there, and then we can process that through something like ImageJ, which is a free application from NIH to analyze images. And one of the things that it can do is it can give you an area from the image. There's also Python libraries to do this. There's R scripts to do it. There's lots of ways to get at it. But for me, when I measure something with small leaves, I use the big chamber aperture. I put the branch in there, or I put the leaves in there, and then I'll take the bottom off and take a picture. If you're not using the fluorometer, and you're using one of our other clamp style chambers, the chamber and the light source are two separate pieces. And so the light source sets on top of the chamber and affixes with just two screws. And so if I have something that doesn't fill the normal chamber, the normal clamp style chamber, I'll just take the light source off while it's in there and take my picture, and then I'll do the same thing. And those chambers are nice because they actually have rulers scribed around the outside aperture. So if you want to estimate it, or if you're working with linear things, you have a measurement tool built into the chamber. So the issue is that you have to know that leaf area. And if you measure it after you collect the gas exchange data, then it's very easy. There's a column in the data file that says area. And if you open the file up in Excel, that's what it'll open in. And you type the correct leaf area in the row, then the carbon assimilation rate and the conductance and the CI and the transpiration rate and all of those parameters that depend on area will get recomputed for the right leaf area. And so it's just like measuring things with big leaves. You just have an extra step in the process that you have to go through.
Okay, Jason. Next question.
Mm-hmm.
Could we conduct temperature curve by Li-Cor 6800? And what minimum and maximum chamber temperature could be controlled by this equipment?
Okay, yeah, that's wonderful. So the 6800, as we saw tonight, has integrated temperature control. And I was controlling at about ambient. That temperature control, we specify it as plus or minus 10 degrees C from ambient. So in my room here, it's a little warm. It's 25 degrees C about. And so my instrument will be guaranteed to go down to 15 degrees C and up to 35 degrees C in that situation. That's the specification. In general, they're better at heating than they are cooling. And so while we say plus or minus 10, it's really minus 10 and plus something greater than 10. It's quite likely that I could get it to 40 or 45 in here without any issue. And so, yeah, the limitation is whatever your ambient temperature is, right? Because we control on bounds around that, not absolute endpoints. But, you know, you could use the generic response curve that I showed, and you could choose to control on exchanger temperature or leaf temperature, and you could give it the set points, and it can just step through them.
Okay, Jason, this is maybe the last question.
Okay.
There are two kinds of Li-Cor you showed here, namely Li-Cor 6800 and Li-Cor 600.
Yep.
Is Li-Cor 600 more better, isn't it? What is your comment? How many advantages of Li-Cor 600 compare with Li-Cor 6800? And how about the result of measurement? Clear?
Yep.
Yes.
That's good. So one's not better than the other. They're different instruments that do complementary measurements. So I see the pair of them as the most powerful. That's the best, right? So the 600 gives me really quick measurements under whatever the ambient conditions are. But it only measures stomatal conductance and chlorophyll fluorescence. Those are great parameters. I can do a lot with those. The 6800, right? That gives me measurements under any condition I want to create, right? So whereas the 600 is subject to ambient, right? I couldn't measure a light saturated 5 PS2 right now with this instrument because I'm in a dark room. There's no light source. But my 6800, it has a light source. I can set it to anything I want and I can make the measurement, right? The 6800 also measures carbon. The 600 only measures water, right? So I get more parameters from a 6800. The fluorometer in the 600 is pretty basic. Like I said, it does 5 PS2, it does FV over FM, it does electron transport rate. The fluorometer in the 6800 does induction kinetics. It does dark pulses to estimate F naught prime, which gives me more fluorescence parameters. So the way that I see these two instruments are a pair, right? This one I use for rapid screening to find interesting individuals. This one I use to do detailed characterizations of physiology, right? So I might use this instrument to parameterize a crop model, and then I might use this instrument to go out and verify my crop model, right? So one's not better than the other. They're similar, they're complementary, but they do different things. One is faster than the other, right? 5-second data. I'm still blown away by that. Okay.
Okay, this is the one for the last. Which the best method of measurement can be used as plane screening for both equipment? Clear?
Yeah. Well, I guess it depends on what you're screening for. And so when I think about a question like that and a screening scenario, I want to think about, okay, so what is the thing that I'm interested in? Okay, well, maybe I'm interested in something related to water relations and drought stress. Well, if I want to screen a large number of plants to find when drought stress starts, right? Well, a good way to find that is to look at stomatal conductance. And this becomes a really useful tool, right? Because it measures conductance and it measures it quickly. So I can screen a bunch of plants in the field and I can find the onset of drought stress. Well, but that's not the only thing that I might want to screen for, right? Maybe it's temperature tolerance that I want to screen for. Somebody asked in one of the previous questions about doing temperature response curves. Well, I can't do a temperature response measurement with the 600. So if I want to screen plants for maybe their thermal maximum, right, then the instrument that I would use to do that would be a 6800. So it depends on what I want to screen for. You know, what's my research question? What's the mechanism of action that affects that question? And then I can pick an appropriate tool based on that.
Okay, Jason. Mungkin peserta ada tambahan lagi untuk pertanyaan, kami beri kesempatan. Masih ada sedikit waktu. Atau mau langsung bertanya ke Mr. Jason? Baiklah, untuk sementara mungkin Pak Paul sama Pak Kukuh mungkin ada komen sebelum kita close, Pak. Silakan, Pak. Halo, Pak Paul. Pak Kukuh.
Iya Pak, saya rasa cukup kalau dari saya. Udah jelas itu semua. Yang penting mungkin Pak gitu loh Pak.
Oke Pak, diulang Pak.
Halo?
Nah, itu Pak Kukuh tuh, Pak Kukuh.
Iya Pak, silakan Pak. Pak Kukuh.
Saya baru dari toilet Pak. Apa Pak? Sorry Pak. Halo?
Ya Pak. Ya, silakan
Ya.
Pak.
Tentang komentar Pak.
Ya, sebelum
Ya.
close Pak.
Oke, ini.
Halo, suaranya hilang Pak.
Halo?
Ya Pak, masuk.
Nah,
Ya.
kan ada alat yang Porometer 600 itu ya, Li-Cor 600.
Betul.
Menunjang kebersamaan alat dengan fotosintesis Li-Cor 6800 itu ya.
Ya.
Karena memang yang saya alami ini dulunya saya pernah mengukur Porometer tapi kurang begitu stabil, not stable in the field. Yo, nanti Li-Cor 6800 insya Allah nanti bisa melengkapi data yang kita harapkan, terutama untuk seleksi varietas unggul. Halo?
Halo, ya.
Ya, udah Pak.
Iya, masuk Pak. Silakan Pak, lanjut. Masih ada komen?
Udah.
Iya.
Ya.
Oke, Pak. Tutup.
Tutup Pak Kukuh.
Oleh, baik Pak. Sebelum
Halo.
Halo, Bapak Paul, ada komen Pak?
Ya mungkin nanti sebelum tutup kita ada saya ingin mengucapkan terima kasih sama Li-Cor ya Pak, sama Unitama juga, gitu Pak. Mohon di spare waktu gitu loh Pak. Makasih Pak.
Oke, Pak Paul dan Pak Kukuh, mungkin nanti closing langsung aja dari Pak Kukuh, mungkin ada sambutan atau penutupan. Khususnya juga mohon kita ucapkan langsung aja dengan Li-Cor, khususnya dengan Jason.
Sudah selesai semua, apakah belum?
Silakan Pak. Ya Pak, silakan ditutup Pak Kukuh. Langsung juga Jason juga ucapan nanti kita sama-sama terima kasih.
Dulu Pak ya.
Yuk.
Halo.
Ya Pak.
For Li-Cor, Nebraska, USA, for 2 days of valuable presentation. This is important for researcher of plantation industry, especially for Mr. Jason and team and this kind of webinar. And once again, thank you very much.
Thank you.
Kepada PT Unitama, terima kasih, dan semoga akan ada webinar-webinar lain yang kita akan laksanakan bersama UPT. Dan saya sangat berharap kerjasamanya ini tetap berlangsung. Dan yang ketiga, saya mengucapkan terima kasih untuk seluruh peserta yang mengikuti acara ini dan sudah menyediakan. Semoga bermanfaat dan UPT sangat terbuka kepada siapapun atau universitas atau institusi untuk melakukan kolaborasi penelitian dengan kami yang ada di Unila. Terima kasih Pak.
Baik Pak Paul, untuk kita beri kesempatan mungkin dari Li-Cor mewakili. Hello Tim. Hello. Tim. Are you there? Okay. Any closing speech from you?
Yeah, I'd like to thank everyone. It looks like we had about 120 people for the second day today, so a strong showing for day 2. A big thanks to Jason, of course, for his wealth of knowledge. I think this was productive, and the questions coming in were excellent for fueling some conversation. And thanks to both Lampung University, Professor Kukuh, and PT Unitama for putting this together. We would of course rather be with you all in Indonesia. That's our preference, but we're limited right now, just as you are. And this is the next best option. So as soon as we are able to travel internationally again, we will be back to Indonesia on an annual basis, and we look forward to that opportunity. So thank you for joining us.
Okay, thank you. Yes. Pak Kukuh mungkin closing Pak, langsung.
Pak Kukuh.
Ya, Pak Kukuh. Ya Pak, silakan Pak, closing langsung Pak.
Ya, terima kasih yang saya ucapkan. Dan juga for Jason and team, thank you for sharing the knowledge. And in Indonesia again, and directly to show our student how to measure how to use the equipment Li-Cor 6800 and Li-Cor 600. So I'm waiting for you, Mr. Jason and Thank you. See you again.
Excellent.
Oke.
Oke, Pak. Tutup, Pak.
Baiklah, Bapak-bapak dan Ibu-ibu sekalian, selama 2 hari kita sudah setengah hari penuh untuk presentasi ataupun pengetahuan dan pemikiran. Sekali lagi kami atas nama manajemen PT Unitama Analitika Perkasa sebagai distributor Li-Cor di Indonesia, sekali lagi mengucapkan terima kasih atas kerjasama yang baik khususnya dengan Universitas Lampung. Ya. Mudah-mudahan kerjasama ini akan terus berlanjut dan juga dengan pihak-pihak yang lain kita terus welcome untuk mengadakan kerjasama dan lain sebagainya. Oke. Sekali lagi kami mengucapkan terima kasih. Sampai ketemu lagi di lain kesempatan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Makasih Pak Agus.
Sama-sama, makasih Pak Paul.
Bagus.
Iya Pak Kukuh.
Ya, kita ngobrol nih tentang RAPP itu.
Oh iya, lanjut Pak. Lanjut RAPP. Mungkin
Memang RAPP dipegang langsung oleh Anderson, Pak.
Iya.
Ya, yang Lydia yang perempuannya itu kakak perempuannya megang sawit.
Asal Negeri Pak ya.
Ya, jadi ada 2 itu.
Iya.
Jadi ya, nanti kalau Anderson kan kalau udah memutuskan seneng alat ini langsung dia beli dia Andersonnya itu.
Luar biasa Pak, luar biasa RAPP di hadapan Li-Cor Pak. Itu luar biasa karena secara sedunia ini Pak tidak ada customer swasta ya Pak ya. Tidak ada customer swasta yang punya tower itu 4 Pak.
Iya.
Tidak ada kalau universitas, mungkin lembaga riset pemerintah ya Pak ya. Itu bisa dimaklumi ya Pak ya pendanaannya. Tapi kalau untuk swasta tidak ada yang sebanyak.
Lumayan itu ya.
Iya, luar biasa bagi mereka Pak. Dan juga mereka sudah publish paper juga kan Pak? RAPP. Mereka udah ada paper yang dipublish juga ke internasional. Iya. Nanti saya akan share ke Pak Kukuh papernya.
Oke. Ya. Apa itu? Butuh Pak Agus butuh email saya? Enggak.
Oh,
nanti
butuh
Pak Paul
loh,
aja ya.
Betul.
Ya, Pak Paul ya.
Iya Pak. Tapi Pak Kukuh juga belum pernah lakukan riset edikovarian ya Pak?
Putus-putus Pak, suaranya putus-putus.
Pak Kukuh sendiri sudah pernah ada pengalaman dengan edikovarian Pak?
Belum kayaknya Pak.
Pak, belum itu Pak.
Belum ya?
Saya lagi nanti Pak Kukuh itu untuk itu minta ke Asian Agri itu Pak.
Ya Pak, biar Universitas Lampung bisa jadi paling maju se-Sumatera nih Pak.
Iya bisa.
Karena posisi Lampung enak deket Jakarta Pak.
Kurang bagus nih jaringannya nih.
Nggak kedengeran ya Pak Paul?
Enggak Pak, nggak putus-putus.
Ya maksud saya biar Universitas Lampung jadi universitas riset yang paling maju Pak, se-Sumatera Pak.
Iya Pak, itu yang diharapkan.
Karena posisinya
kita juga mohon dukungan dari.
Iya Pak, ya kan kami sudah mendukung Universitas Lampung dari tahun 2007 kan Pak?
Iya, terus lah Pak ya, jangan kapok-kapok kayak gitu loh Pak.
Iya belum pernah kami kapok Pak. Karena bagi kami kan yang penting Bapak peneliti bisa lakukan penelitian, dan mengenai misalkan ada masalah, perlu perbaikan, kan bagi kami kan Bapak silakan dipakai dulu alatnya, pembayaran bisa dilakukan tahun depan kan Pak, bagi kami tidak masalah.
Ya, selama ini kita lakukan itu Pak.
Iya.
Halo.
Iya Pak Paul.
Nah, ini agak buruk nih jaringannya, ini sering putus-putus ini. Mungkin di sini Pak itu masalahnya.
Iya. Untuk ini Pak, untuk yang pembacaan yang mungkin masih perlu dibantu oleh Li-Cor, nanti silakan Pak, nanti kita bisa atur
Email ya.
lewat email atau mau video call juga bisa Pak.
Oke,
Li-Cor akan
oke.
supaya pembacaannya bisa lebih cepat menghasilkan paper lagi ya Pak, selain yang sekarang
Iya,
sudah
iya.
ada. Sekarang sudah berapa paper Pak keluar Pak?
Udah banyak dia tuh Pak.
Oh, udah banyak.
Sudah.
Berbasis 6.800 Pak ya?
Yang 6.800 kan kita baru Pak. Kalau dia masih yang 6.200 itu dulu.
Oh iya. Ya, 6.800. Ya, mungkin yang paling banyak mengukur Pak dengan 6.800 itu yang di Kebun Raya Bogor Pak. Karena mereka sampai rebutan Pak. Rebutan antara LIPI dan Kebun Rayanya. Karena alat cuma satu. Mereka bilang itu kan dana bantuan dari JICA Pak, mereka itu.
Ya, betul.
Akhirnya mereka minta Fluorometer juga mereka upgrade Pak ya. Nah, setelah itu mereka kan belinya tanpa Fluorometernya. Akhirnya mereka dapat dana lagi, dibelikan oleh JICA lagi untuk upgrade ke Fluorometernya, Pak.
Iya. Ya mungkin saya ke depannya itu yang Li600 itu dulu Pak. Untuk melengkapi
Nah,
yang
Li600 Pak, ini demo unit kami akan datang minggu depan.
Ya.
Saya tawarkan ke Unila
Ya.
untuk coba pakai dulu. Silakan Pak, nanti kami kirimkan. Masih tes Pak.
Ya, nggak apa-apa Pak. Kita mah sabar menunggu.
Kalau gratis ya sabar.
Nanti juga kalau nanti dari peserta lain kalau ada yang mau coba 6.000 seri 600 ini mungkin juga nanti bisa kami tawarkan juga Pak.
Ya, silakan Pak.
Setelah dari Unila maksudnya.
Ya, silakan aja Pak. Tapi ya kami berusaha untuk ini, untuk melengkapi yang 6.800 ini Pak nanti. Nah itu Pak yang masalah ininya Pak, chambernya ya.
Halo Pak Paul.
Halo, kedengaran nggak Pak?
Ya.
Ya. Kalau chamber Bapak tinggal lengkapi ya Pak?
Iya, itu maksudnya.
Ya.
Agak kesulitan kalau yang sekarang ini.
Yang itu Pak, yang mungkin kalau Bapak ada penelitian ke soil flux juga, kan aksesoris untuk soil fluxnya Pak.
Iya. Nah itu masalahnya kan.
Nah kalau ini Pak, kebetulan soil fluxnya itu, soil chambernya kami punya Pak. Kalau nanti sekalian mau ada percobaan penelitian, kami bisa pinjamkan juga Pak soil flux chambernya Pak.
Oke. Terima kasih banyak itu Pak. Tapi ya kita ya nggak enak juga lah minjem terus Mas. Maksudnya kalau bisa dilengkapin ya lengkapin aja sekalian kayak gitu loh Pak.
Ya maksudnya coba dulu gitu. Oh ternyata memang butuh nih ya, silakan Pak diusahakan nanti.
Oke Pak. Terima kasih Pak masalah itu Pak.
Selama jaraknya kan masih memungkinkan kita bepergian Pak ke Lampung.
Ya. Karena masih dekat.
Iya.
Yeah.
Oke, mungkin begitu Pak. Kalau ada lagi nanti
Halo.
yang lain.
Mudah-mudahan universitas lain itu mulailah menginvestasikan yang hal-hal yang penting gitu loh Pak.
Yang penting iya, ya. Nah masalahnya kan namanya universitas kan banyak jurusan Pak, saling berebutan Pak.
Iya, apalagi lagi COVID ini udah saya usulkan itu di pimpinan itu. Ya udah dibelanjain aja. COVID ini kan masalahnya kegiatan belajar daring. Aduh. Halo?
Halo Pak Paul, suaranya putus
Iya.
Pak.
Iya, memang ini lagi jaringannya kurang bagus. Nanti kita WA aja, Pak, ya.
Oke.
Nah, biar lebih lancar, ini jaringannya nggak bagus, Pak.
Ya. Terima kasih semua kepada para peserta. Sekali lagi, Pak Kukuh, Pak Paul. Sampai ketemu di kesempatan lain. Selamat siang semua. Thank you, Jason and team, for your support. Bye. Have a nice day.